points when none of us died. You're going to be fine. And then this year, when I took them for the scientific writing workshop, and we are dying. We're not going to make it. And I'm looking at all of you here. How many of you died? Can I see the hands of those who died? Can I see the hands of those who died? Guys, it's called summiting Mount Everest. Closer to the top, you're going to start feeling like you're losing your energy, you're losing your oxygen, you're losing everything, but you are close to the top of the mountain. And this is where we are today. Beginning of the year, I'm sure you didn't even see this day coming, but today you are here. And you are ready to present your projects to an audience like this one. So guys, clap hands for yourselves. You are here now. You are here now and you didn't die. You are still alive. Before we start with the welcoming, this class that you see in front of you is the COVID. These are our COVID babies. They joined us in 2020, just before COVID, and they've experienced the ups and downs of all the changes that had to be made to accommodate, educating students during a pandemic. So they can write a book about the journey. And one of the things that I want us to acknowledge is the fact that they were first years when they had to go through the traumas of losing parents, the traumas of losing family members, and they were, and we were expecting them to perform at school, and we were also expecting them to moan at the same time. And not only just them, but all of us. So I would really like for us just to take a moment of silence. I know we're from different religions. You can say a prayer to, according to your religion, just a moment, 1 minute of silence, and to just remember those we have lost in the past 3 years, because, you started just before the pandemic, and there was a lot of roller coaster feelings happening that year. So let's take a moment and remember those that couldn't get an opportunity to watch you develop to who you are today since you started this journey. Just 1 minute, moment of silence. Thank you. Thank you, colleagues. Thank you so much. Our HOD was supposed to be here to welcome you all, but life is unpredictable. She really wasn't well when I spoke with her this morning, so I'm going to take over her duty for the day and do the welcoming. She would have loved to be here to greet all of you as the head of the department. First and foremost, I'd really love to welcome our students. We are here because of you. How many third-year students that are here? Do we have any third-year students, or they're busy cramming for the test somewhere? We've got one. We would have loved to have more third-year students, because next year they are going to be on the hot seat. A warm welcome to our fourth-year students who have been working hard, trying to apply the principles of research and doing their projects. Welcome, fourth-year students. A warm welcome to our academics and supervisors who have also been working hard trying to make sure that our students present the best of the best projects at the end. So welcome, colleagues. We also have supervisors that are not from the Department of Pharmacy. They came in and they said, listen, we also want to make a contribution. We've got Dr. Mpo from Nanosciences, who has been helping together with Dr. Mufamadi. They've been working as a team they brought in the nanotechnology into our fourth-year project. And we also have Ms. Donne Kritzinger from Biochemistry, who specializes in pharmacodynamic projects, and I also want to welcome her as one of the supervisors. And a warm welcome to the Faculty of Health Science Research Directorate is here. We've got Prof. Melariri over there in the corner, who's the Director of Research. Welcome, Prof. It's good to have you in our midst. We also have her team with us, Ms. Tembeka Sidinane. When you do your postgraduate studies, you're going to know who she is. She's one of the engines there when it comes to, the projects that we do, the postgraduate projects. Welcome, Tembeka. Bethlyn, where's Bethlyn? I saw her. Hi, Bethlyn. Bethlyn from Ocean Science. She's also here. One of the members of the research directorate. Zuki, I'm not sure if Zuki is here, and Lutando is here. Welcome, there's Zuki, I see you. Welcome all of you. These are the people that deal with the research, the faculty, our pharmacy research. They are the ones that handle it. They handle it like a precious cargo. So we have to welcome them and give them some love. We've got our judges, the most important people here, because if there wasn't going to be judges, I'm sure half of you wouldn't be here. Or you wouldn't have put in enough effort in preparing your presentations. We've got Dr. Rian van der Venter from the Department of Radiography, right? Am I saying that we were told we must say it's not the radiology department, it's the radiography department. And then Ms. Tembeka also is one of our judges from the research directorate offices. And then we've got our guest speaker, Ms. Maxine Tromp, who is coming in and who's going to be talking to our students. And guys, please pay attention. You've got important people here that are here to speak to you. So I'm sure, Maxine has cooked up some nice oxtail for you. So make sure your appetite is nicely enticed this afternoon. Also want to acknowledge the presence of our veteran, one of my favorite people in this world, Prof. Naidu. I've known Prof since I was an undergraduate student. I was not a pharmacy student, but I knew him. And he's been one of the biggest supporter of pharmacy research, both undergraduate and postgraduate. So he always supports us. And if you want to know anything, I always say when I joke, he is like a mobile library. You go and you search in his mind and you'll find what you are looking for. That's the amount of wealth he has of the knowledge in this field. Also want to acknowledge, I think I see a very beautiful lady there, who is Ms. Latechan, who is joining us. Thank you so much for coming in. You know, I always, when I see her, she reminds me of my hometown, because she was a pharmacist there back in the day. So I always smile when I see your face. Thank you so much for joining us. I'm not sure who I am leaving behind. I'm not sure if we've got representatives we've invited guests from Adcock and Aspen. Aspen, as you have seen, Aspen is one of our sponsors. So if I have missed them by mistake, my apologies. I'm trying to look through the crowds, but I don't seem to be spotting someone. But as the time goes by, I will acknowledge the presence of people. So thank you so much, colleagues. That sums up my welcoming. I hope I didn't leave anyone behind. Thank you very much. I'm now going to hand over to Mrs. Perry, who's going to introduce our guest speaker.
Good afternoon, everybody. Welcome. We've had the welcoming already, but I've got the wonderful task of introducing our guest speaker today, Mrs. Maxine Tromp. Mrs. Tromp is Maxine. I think I'll call you Maxine. Maxine is an alumni of this university. She finished her BPharm in 2015, so not so long ago. At that stage, it was NMMU. She did her master's after that and graduated with her master's in 2019 at Nelson Mandela University. She then also has done the PMART and the PCDT courses, which you may have heard of. Currently, Maxine is the compliance manager of the Clinicare Pharmacy Group. I'm sure you've all heard of Clinicare. She has also worked in various sectors, community, corporate and independent, as well as institutional sector. Maxine, her next journey is just about to start, she tells me. She's hoping to do a PhD at NMU, so we hope to be part of her next journey. And it's all for me basically to say that I think that Maxine is going to be able to share a lot with you. I think you will learn a lot from her in a short time, because I really think you guys are going to be able to relate to her. Not like me, that finished a very long time ago. You've now got somebody in front of you that actually remembers sitting exactly where you are now and the journey going forward for you, where you're going. So thank you, Maxine, and please come and join us.
Right, I'm sure everyone can hear me. Good afternoon, everyone. Thank you for inviting me here today. I must say it's quite a nostalgic feeling, as I sat exactly where you guys are about 8 years ago with Mr. Masebe being our supervisor. My hands were sweating. I didn't know if our presentation was ready. So good luck for today. I am quite excited to see the projects that come through. So to recap quickly, I completed my internship as well as my community service in a community pharmacy setting. And this is where I fell in love with people, patients, being in a clinic, and later on I focused more on a managerial role. Being drawn towards clinical work, I then enrolled and completed the PMART program as well as the PCDT course through Northwest University. During my first year of PCDT training through Northwest is where my passion for research actually developed. I became quite intrigued in this particular field that I was now learning about, and basically where these pharmacists were practicing in South Africa and what was happening in the environment. Now, why am I telling you this? I was never drawn to an academic career. I didn't see myself lecturing. I love people. I love patients. I want to be in the clinic. So doing a postgraduate study was not necessarily something that I was always drawn to during my undergrad. So completing a master's degree, yes, it will add to your academic achievements and your resume, but it's the journey of the master's and it's the journey of a postgraduate that I'm actually here to talk to you about today, and what you can gain out of it for any sector of pharmacy that you are going to possibly find yourself in one day. I also got notes. So during your projects, I know Dr. Dambuza alluded to scientific writing. You guys have been guided by your supervisors and your colleagues and your other students about your referencing and your graphs and your figures and the style of your project. And I'm also quite positive your project was returned more than once to say, can you please have a more scientific style of writing? Now, scientific writing, which I've now learned, is not only about proposals, about articles, and about projects, but it's actually about professionalism. Having a fine-tuned writing skill prepares you to respond to other medical professionals. On a daily basis, I have doctors, as I'm still quite involved in the clinical side, asking for a comparison of 2 drugs for 2 different patient profiles. In order to respond professionally, I need to have a refined writing skill. This also, in a community pharmacy setting, helps you to write referral letters for your patients back into the government system or back into private sector. On a completely different topic where I'm also quite involved in is wholesale pharmacy, a little bit of manufacturing pharmacy, and medical devices. So almost every day I deal with South African Pharmacy Council and SAHPRA when it comes to improvement plans, inspection reports, and audits. Being able to respond to these requests from these governing bodies in a scientific manner is incredibly important. So having a refined writing skill will give you the opportunity to adequately and successfully apply and maintain any pharmacy license which you might own one day. So as we've also spoken about how the students felt like they were all dying, a BPharm degree is a demanding 4 years. You are expected to have organizational skills and good time management. These skills are the bare minimum that your future employer will expect from you one day. By completing a postgraduate degree, your organizational skills and time management becomes incredibly enhanced. And this is in plain sight when your prospective employer is sitting with 100 CVs on his desk and yours is the one that has a postgraduate qualification in it. Thirdly, and quite interestingly in the last, because you guys are the COVID babies, learning how to recognize information which is truthful, relevant, and current. So completing a postgraduate degree teaches you how to look for information that is trustworthy. Any healthcare worker will understand how critical this is over the last 3 years. Apart from the pandemic and the information that was floating around on social media, these skills are often used when it comes to medicine recalls that you will see in any form of pharmacy, as well as counterfeit stock. So in South Africa, counterfeit stock is something that we also see from time to time, and just to be able to differentiate information that is truthful and current versus information that doesn't actually carry a lot of weight. Then in my opinion, one of the all-time perks of pursuing a postgraduate degree is making contacts. So sitting around this room today, you will all know your fellow students and your colleagues and your supervisors. But pharmacy is an incredibly diverse profession. Once you get out into the world, you will notice that as diverse it is, it's an incredibly small profession. I walked in here today and I see supervisors and lecturers from when I was at varsity. I see pharmacy students who I've worked with. It's an incredibly close-knit environment. If you do pursue a postgraduate study, most of your studies will have data collections in the form of questionnaires or expert focus group discussions or editing. You will meet pharmacists and other experts in South Africa that have the exact same interest in the field as you. Following on this point, if my memory serves correct, your fourth year topics that you guys have just researched are topics that are given to you. So I think we could choose like 3, and then it was kind of a pull out of a hat which topic you got. So during a postgraduate, however, you can actually investigate your own interest, which is exactly what I did with my master's, and it's what I hope to do with a PhD. So my passion is clinical, it's PCDT, and it's community pharmacy. It's not theoretically an academic setting. If you found yourself not drawn to your current topic, I can understand that research feels like an absolute mountain to climb. However, you can, I guarantee you, if you research something you are passionate about and you believe it can benefit your career, you will have the passion to continue and complete the study, because I know just how hard it is. Every time you get something back, there's a red pen over it, you really don't want to continue. But if you are interested in it, you will have the passion to carry on. So just to recap, studying a relevant postgraduate course with your interests will increase your level of specialist knowledge, and that is compared to any other undergraduate. This is a marketable benefit if you are applying for the same job as somebody else. You'll have a chance to engage with experts, colleagues, and companies during your research, opening up opportunities that might not have been available to you beforehand. And you will position yourself as an expert in your field. So before I end, I hope I made pursuing a postgraduate study a little bit more interesting. However, you will also learn some important life skills. So back when I was completing my BPharm, in first year, we had a subject called WRFE 101. Does it still exist? So this is now Computers 101. First lecture, you get there, turn the computer on, use the mouse. Little did I know, I should have attended more of these lectures. So fast forward to 6 years, I am at the start of my master's journey and I was busy writing my proposal. Now I don't actually see my supervisor here today, but I think she would have a bit of a chuckle because I don't think she knows this. So the topic I research was PCDT, and there's very little published articles in South Africa at the moment. So I used as many articles as I could to try and justify my thought processes. So every time my supervisor sent me my proposal back, there was these big red marks. There was more red on my proposal than anything else, with big circles all over my paragraph saying, please justify, and like these open brackets that says, please justify. So each time I went back to the drawing board, I was looking for more articles. I'm like back in 1990 looking for articles on PCDT, and I just wasn't winning. So during a postgraduate, you will have many breakdowns, many early morning coffees, late night wine, lots of tears. And I was busy chatting to one of my colleagues at the time, and I was like, this is horrible. I'm going to quit. I think I'm like 4 months into I haven't even finished my proposal, and it was just a horror. And she looked at me and she said to me, did you control J your writing? I said, what are you talking about? She said, on Microsoft Word, that thing that says justify, that makes your writing look all very pretty and crisp. So needless to say, my referencing wasn't the problem. I should have just gone back to WRFE 101. So apart from becoming an expert in your field, you will also become an expert in the basics. So in closing, broaden your thoughts on a postgraduate. It's more than academia. It's a gateway to opportunities within your field of interest. If your fourth year project sparked any interest in a field, I encourage you to pursue it. Our fourth year project actually led to one of my group members finishing a master's degree in their fourth year project. And I see some of you are still, I won't tell you which project it was, but still broadening on that topic, which I think was the first time that topic was done in 2015. So thank you to the Pharmacy Department again for inviting me here today. I hope I was able to entice even one or two of you to pursue a postgraduate career. Thank you.
I'm sure you have questions for Maxine. I've asked her to stand next to me. She said the very funny things. I was listening and I'm thinking, oh gosh, I hope you guys are taking a lot of notes here. Any questions for Maxine? Guys, there's a question from Prof.
Thank you very much for your speech. I just want to find out, you mentioned counterfeit medication.
Yes.
Which you come across every now and then. Can you just tell us how you identify that, or do you get most of the time information from the pharmaceutical companies saying that this batch is not our batch, it's a counterfeit one? So I was just curious about that. Thank you.
So quite an interesting topic, because it often starts out that it's always a Viagra that comes in somewhere. So we do often get hints either from SAHPRA or South African Pharmacy Council to have a look, and especially on your originator products, whether it's, for example, Viagra, Pfizer, a lot of our boxes in South Africa actually either have a QR code on them now, or they are almost embossed with almost like a shimmer, like on our banknotes. So often the counterfeit medication doesn't have this telltale embossing on it or the shadow like you would see with our money. So that's kind of where it starts. It's generally, it will come through from SAHPRA or from South African Pharmacy Council, and it's often something that's imported. It comes through the port, so whether Durban, Cape Town, or in our Kruger Harbour, and it makes its way onto our market. So yeah, I hope that answers.
Any questions from the students? How many of you want to do postgraduate studies in the future? Not necessarily next year, but in the future. Yes, any questions? What do you want to know? Not
yes.
How do you find the social impact of doing BPharm in masters?
Did you say emotional impact? You should probably ask my husband that, but or like the impact on the family. So your BPharm is very structured. You've got your modules. You are told what you need to finish by what time. A master's or postgraduate, they throw you in the deep end. I mean, we love our supervisors, but you are a professional by the time you do a postgrad. So again, I was very motivated about my topic because it was something that interests me. My master's was, to me, easier than my undergrad. But I'm sure some of you who have done your projects found it like quite, oh, I have to do this now, and I have to do this, and you just want to get the boxes ticked. And then I can imagine it being very draining. So it also depends. I mean, there are a couple postgraduate opportunities that are course-based and not research-based. So I know there's a manufacturing one, there's clinical pharmacy, and those are more course-based intense with a smaller research component where something like a PhD here or a research master's is a full research component with no course-based material.
Okay. Thank you very much. Thank you. Thank you so much, Maxine. I think what we can, when we talk about postgraduate, we gave you projects. Some of you probably didn't even like the projects we gave you. They're not the ones that you chose, but you're going to be given an opportunity now to be in the real field, in the real world, and you will probably need to, you will probably need to learn something new, identify something that interests you, and that will give you an opportunity now to bring in what Maxine talked about in terms of postgraduate studies. Now you choosing your own project, you're finding your niche, and you are going to do a project with a topic that really attracted you, not the one that was dished out to you by your supervisors. So thank you so much, Maxine, for that, and I hope you've been enticed. I hope the oxtail was really nice and tasty, and I hope in the near future to get emails from you inquiring about postgraduate studies. We've got a few that have come through, and we welcome you, and hope that when you've settled in the field and you've identified the area that interests you, we will see more applications coming through. So now we are moving now to the real reason we are here, and that is, so I need to find where are my presentations, ZRE presentations. We're going to start with the development of evaluation of sublingual vitamin D dispersible films. Is that team here? You can start.
So as mentioned earlier on, we're working on the development and evaluation of sublingual wafers. These are also called oral dispersible films and abbreviated ODFs. And Christabel was working with me, and our supervisors were Dr. Luvino Kili and Mr. Ngomo.
So as mentioned earlier, this was a ZRE400 project that focused on the study and development of sublingual 600 international units vitamin D3 wafers. So vitamin D, it's a fat-soluble vitamin, and it is mainly found in two forms, which are vitamin D2 and vitamin D3. And in this study, we're focusing on supplementing vitamin D2 instead of vitamin D3 instead of vitamin D2, because vitamin D3 is more efficacious in raising serum vitamin D levels than vitamin D2, and also it has a longer duration of action. So vitamin D deficiency causes a lot of health problems, which include bone diseases like osteoporosis, osteomalacia, and rickets, and also causes cardiovascular diseases, and it also compromises the immune system, predisposing the patient to viral and bacterial infections. So it is so important that we give an effective route for supplementation so that the drug can reach its targeted site of action, and the drug dosage has to be tailored for patients with also special needs. So the problem statement was that there are a lot of dosage forms that are already there on the market, injectables and tablets, but this cannot tailor for people with needle phobia, dysphagia, and also malabsorptive issues. So we're like, let's try to find a dosage form that can tailor for those special needs. So the aim of the study now came to be formulating and evaluating sublingual vitamin D3 wafers as an effective dosage form for daily supplementation. And objectives were to conduct a pre-formulation study to determine the optimal formulation of vitamin D3 sublingual wafers, determine the optimal manufacturing method of the vitamin D3 sublingual wafers, to formulate the actual sublingual wafer, and then conduct quality control tests.
Moving on to the methodology, for incorporation into an oral dispersible film, the API needed to have a low molecular weight, good solubility. It needed to be lipophilic so that it can penetrate into the mucosa, and it also had to have a good taste, or rather just be bland. And vitamin D3 met all these requirements. It was bland, it is lipophilic, and it is a low molecular weight. And our method of formulation was solvent casting method. We had various methods that we could have employed. However, hot melt extrusion could not be used because it was not compatible with our API that is heat sensitive. And then rolling method was not feasible in the lab as the cast was so watery, hence it could not be rolled. We then settled for solvent casting method. And we found that it produces flexible wafers with better content uniformity, and they also have a glassy feel, which makes it aesthetically acceptable. And in the materials, if you look at the table, you will see that we had our API, film-forming polymers, and going down to the vehicles. So we were giving preference to excipients that have double roles. For example, the extra virgin olive oil was working as a penetration enhancer and a surfactant, and then ascorbic acid was working as an antioxidant and a saliva stimulating agent. This was important for us as our film is so small and we require to use the minimum number of excipients as possible. So this is our manufacturing process flow, where we first of all made two separate mucilages with the film-forming polymer and then the sodium alginate, and then mixed the two in a mortar and with the pesto, and then dispersed the other solid ingredients, that is the sucralose, ascorbic acid, and colorant. And then we then added a polyvinyl acetate, and then we separately dissolved our API in a mixture of oils. As you know that vitamin D3 is lipophilic, and then we then cast this mixture onto a glass towel, and then we placed it in a preheated oven for variable times, as you will see when we talk about our batches, and then we then obtained our wafer product, to which we then cut into the desired sizes. So the batches made, we first of all made placebo 1, which was starch-based wafer, because if you look at all this, you'll see that we were dealing with three film-forming polymers, and we wanted to make a choice whether we are going to use one or more. And then we found that the starch-based wafers that we made in placebo 1 were not favorable, as they were easy to break. And then we then moved on to make a placebo 2 with methyl cellulose as the film-forming polymer. Unfortunately, it was difficult to work with as it formed a lot of bubbles, and it was difficult to work with. It was too sticky. We then moved on to make product 1, as we thought since placebo 1, the only issue was breakage, we then could make product number 1. Unfortunately, when we added our colorant, it formed a brown sediment at the bottom, and this was undesirable. And we also noticed that it was moist. Unfortunately, when we tried weighing, we could not find how much weight it had gained from the exposure to light and moisture. So we had to also extend the period of time from just 3 days to 30 days, so that we can actually see how much it had gained due to the sensitivity of the scale just being 0.1 milligrams. So we then found that in 30 days, it had gained 0.01% mass gain, which then meant that from the third day that we could not weigh it due to the sensitivity of the scale, it had gained 0.01%. And then we then moved on to product number 2, maltodextrin-based wafers with the yellow colorant. Unfortunately, this was not desirable film-forming polymer, as it rolled easily and it was too sticky. It was difficult to scrape off the towel. We then tried to make product 3 by removing the coloring, to which we had problems in product number 1. Unfortunately, it was breaking when we were trying to scrape off the towel. We then had to stop and go back to making placebos, to which we made placebo-based wafers, and we added PVP, which was acting as a filler. Unfortunately, it just did not work with maltodextrin. It remained difficult to scrape off the towel. And then placebo 4 was successful with our starch, and we found that it was now very easy to scrape off the towel, and it had a good folding endurance, which then pushed us to then formulate our final product, which was starch-based wafers with the PVP and no colorant.
So those are the wafers that we made as our final product, which were 1 by 1 centimeters dimension and an average mass of 35 milligrams. So we conducted tests in these wafers, and the disintegration test was 35 seconds. Friability was 0% weight loss. Swelling properties and folding endurance were also acceptable according to the sources that we're using. And these two properties, swelling properties and folding endurance, can actually be modified by just adding or increasing the amount of sodium alginate or plasticizer in the wafers. We also conducted dissolution studies, and we saw that 85% of the API was released within the 15 minutes, which showed that our wafers were fast dissolving. And we also conducted uniformity tests. There was variation of the API in the wafers, but still they were still within expected limits.
So in conclusion, we can see that vitamin D3 containing sublingual wafers are actually a viable dosage form that we can consider in future pharmaceutical formulations. And hence, this research project could be used to then further all the studies and formulate a vitamin D3 sublingual wafer.
Any questions for this group?
Thank you.
There's a question, Prof. Wallenstein.
Thank you, Christabel, and benevolent. Good names. Very interesting project. I wanted to ask you, when you did your disintegration time and dissolution testing, what medium did you use to evaluate that?
For the disintegration test, we just used water because we thought since saliva and water kind of have the same properties, so there won't be any difference in the time, in the quality of the medium that we're using. And for the dissolution test, we used it in a sodium lauryl sulfate-based medium, but the pH was 6.8, which is kind of like the same pH that we can find in the mouth.
Thank you for a very good presentation and the time you've taken to stand there. My question, I think, is twofold. I just want to find out, seeing that you're looking at sublingual, why did you think about this dosage form sublingual, and why the route of administration sublingual, and why did you choose a wafer and not any other dosage form?
Okay, to respond to your question, we chose sublingual because we were trying to cater for the other patients who are having issues with malabsorption, the patients who have needle phobia, and the pediatrics who have swallowing difficulties, to which we didn't consider a tablet. And then we also did not formulate a sublingual tablet because the oral films have an advantage of reduced choking risk, and they are also there is increased patient compliance according to the literature that we found online, to which more patients are now preferring the ODFs over the sublingual tablets. The tablets also have hardness and friability issues, which is then why we settled for a wafer that does not have all those issues. Yeah.
One more question, Prof. Borenstein, and vitamin D tablets, get ready. You will be followed by paracetamol.
Thank you for your presentation. I just wanted to find out from a cost perspective, if you compare this dosage form, let's say with tablets, are there any advantages? Because obviously if one works with a dosage form, you also want to take a look at costs to make it, from that perspective, worthwhile for the patient. Thank you.
Okay. Unfortunately, we cannot speak to large scale because this was small scale, but when we were doing it small scale, it was less time consuming. We only spent, I think, 1 hour trying to formulate and put everything through the oven. So I think it would be, and since we're also using excipients that are readily available in the lab, it's quite cheap. So we can hence say on small scale, it's quite a favorable formulation.
For our supervisors, Dr. Luvuno Kile and Mr. Komo. This is the table of contents. Okay. So what is vitamin D? Vitamin D is a fat-soluble vitamin that the body needs for the regulation of calcium and phosphorus, which are needed for bone health. The deficiency of vitamin D causes insufficient bone mineralization, which then causes osteomalacia in adults and rickets in children. Research has also shown that a lack of vitamin D is linked to a risk of diabetes, cancer, cardiovascular conditions, and mental conditions such as the Alzheimer's disease. The deficiency is mostly treated with oral dosage forms of vitamin D, but these do not work for people that are struggling with malabsorptive conditions such as the Crohn's disease and inflammatory bowel syndrome, as well as people that struggle with swallowing tablets. Okay, so this is what gave rise to our problem statement. Our research aimed to develop and evaluate 1,500 international units sublingual vitamin D3 tablets, and the objectives were to formulate sublingual vitamin D3 tablets by first identifying suitable excipients, as well as manufacturing the sublingual vitamin D3 tablets using the direct compression method, and then conducting chemical and physical tests on the tablets that would have been formulated. So the question then becomes, why the sublingual route over the oral dosage forms that are there? The sublingual route bypasses the first-pass hepatic metabolism, which then increases the cholecalciferol serum levels. The sublingual route also has a quicker onset of action and higher bioavailability because of the large ventral surface that it has. And it also improves patient compliance because these tablets are easy to use and are not affected by food.
Good afternoon, everyone. Thank you, Nyasha. So I'll be taking you through the methodology. The picture that you can see on the screen is the Eureka tableting press machine that we use to compress our tablet. Unfortunately, it broke during the process, so the results that we are going to be presenting today are going to be from one batch. So during formulation development, the formulation that you can see on the screen was adapted from literature. So from literature, we initially had 8 formulations, which were reduced to 4 formulations because of excipient availability in the lab. And then from the 4 formulations remaining, we then reduced it to further 2 formulations because of the method of manufacture. As you can see on the table, we had vitamin D3 as our API. However, PEG 600 was replaced with polyethylene glycol 400. Although PEG 200 to 600 had the same physical properties, we didn't have an issue with that. Sorbitol powder was then replaced with mannitol. Sorbitol powder is just a less sweetener than mannitol. And then povidone K25 was replaced with povidone K30. And then the weight of our tablets was 150 milligrams. So we used the direct compression method as the method of manufacture in our research study. So what we did is we dissolved the vitamin D3 in PEG 400 as vitamin D3 is insoluble in water. We then melted PEG 1500 at 45 degrees and then we added it onto the first step. We then let the mixture cool down to 25 degrees Celsius and then we passed it through a 425 micrometer mesh screen. We then added lactose monohydrate and then we passed it through a 600 micrometer mesh screen. We sieved the remaining excipients excluding magnesium stearate via the 800 micrometer mesh screen and we mixed it with step 4 for 15 minutes. After 15 minutes, we then added magnesium stearate and we mixed it for 5 minutes and then we compressed the tablet. The picture that you can see now is the powder before and the tablets after compression. So the results from our study showed that our tablet ranged between 130 milligrams to 200 milligrams per tablet, with a tablet average of 150 milligram. Our standard deviation then showed that there is a plus minus 20 milligram variation from the 150 milligram average in the tablet weight. We then did a disintegration test, and according to the USP1 of 2019, for a sublingual tablet, it follows the uncoded tablet route. So if 6 tablets are placed into the disintegration machine and 6 tablets disintegrate within 15 minutes, the tablets then meet the disintegration spec. We disintegrated 6 tablets and our average disintegration time was 5 minutes and 40 seconds, which met the disintegration spec. We then did UV spectrometry and then we realized that API content was not evenly distributed within the tablets, and this could be attributed to the powder blending manually done instead of using the powder blending machine. We then did the hardness test using the Eureka hardness machine, and tablet hardness for sublingual tablets is determined by the tablet monograph, and this is found from literature. We then found that our tablets are within the hardness spec. We then conducted a friability test, and there was a 0% friability, 0% tablet loss, and then the mechanical resistance of our tablet is acceptable. Our limitations in this study was time and lab resources, such as the compression machine breaking before we could press the second batch. In conclusion, we aimed to develop and evaluate a sublingual vitamin D3 tablet using suitable excipients and the direct compression method as a method of manufacture. And in our work, the sublingual vitamin D3 was successfully developed and our objectives were met. We then concluded that the results from our study show that there is a very promising potential of the sublingual route as the route of administration, as it bypasses the first-pass hepatic metabolism and could potentially increase the bioavailability of the drug. A famous quote from Eleanor Roosevelt said, "The future belongs to those who believe in the beauty of their dreams." Our dreams could not have been met without the following people: Dr. Luvunokile, who is not here today, Mr. Ngomo, who is here today, for their tireless efforts, support, guidance throughout this project, all the Mondays we had, to Mrs. Jean van Jaarsveld from the pharmaceutical lab and Mrs. Arista van Jaarsveld from the chemistry lab. They allowed us to utilize their equipment and held our hands throughout this project. We thank you.
Thank you for a nice project. I just wanted to ask, and I know you've got limitations in the lab with your tablet compression machine and the size of it. Did the weight variation worry you? Do you think it was not too wide? And then the second part I wanted to ask you is the shape of the tablet. But there too you might be limited with the tooling facilities that you have. Because for a sublingual tablet, I think what I saw in the photograph was a little bit too large.
The weight variation, is it a problem or not? Should I answer now?
Okay, I think all of those were attributed to the punch and die set that we used. So all of the problems that we also had with the weight variability were attributed to the machine that we used. Yes.
Thank you, students. That was a lovely presentation. I want to ask you questions, maybe a bit unfair, because it's going back to the previous group as well. I didn't get to ask my question, but since you studied vitamin D, you might be able to answer it. The previous group said before they chose their solvent casting method, they rejected a method because vitamin D, they said, was sensitive to heat, but yet both in their method and your method, you heated it up, I think, to 45 degrees, the previous group to 50 degrees. So can you tell me up to what temperature is vitamin D heat sensitive?
To answer your question, we did not heat up the vitamin D. We heated up the polyethylene glycol, and then we mixed it with the polyethylene glycol. We heated the polyethylene glycol 1500, and then we mixed it with the polyethylene glycol 400, which already had the vitamin D. No, it was at 45 degrees, so it was not hot. It was just because the polyethylene glycol comes in crystals, so we had to melt those. It was not hot, it was just warm to touch.
One more question. While the paracetamol group, we might as well keep the pharmaceutics in one session.
In most cases, we would see the sublinguals being used for emergencies when patients are, in emergency cases. And can you justify the development of the D3 in emergency cases, saying that you have chosen the sublingual? Thank you.
Okay, so we formulated the vitamin D3. We have vitamin D2 and D3. So we formulated the D3 because it is much more potent than the vitamin D2.
And also, because of the literature study we did, the deficiency of vitamin D3 was linked to multiple diseases. So we want, in terms of the sublingual route, we wanted it as a daily supplementation for patients. So because the normal traditional route is very non-adherent. So we wanted something that is easy and efficacious, and also that you just put it under your tongue and you just go about your day. So that's why we ended up with the sublingual route instead of just the oral traditional route.
Good afternoon to everyone. So we are the ZRE 400 group that was in charge of synthesis and formulation of paracetamol chewable tablets using quality by design. These are my group mates, future quality assurance officers at Aspen and Adcock, and then those are our supervisors. So there is our table of contents. And then when it comes to the background of paracetamol, it is an analgesic and an antipyretic drug that remains one of the most utilized over-the-counter medications in the world. It is available in various dosage forms such as Elijah's, hard tablets, suppositories, and it is also offered at different strengths. So then, when we move on to chewable tablets, they are an oral dosage form that requires chewing before swallowing the chewed particles. These tablets are formulated in such a way that facilitates easy chewing to release the API and then swallowed. So then, they offer a wide margin of safety, and they allow for easy use in different population groups, from children to elderly individuals, especially ones who have a difficult time swallowing traditional dosage forms. And then we move on to quality by design. So with quality by design, it is a systemic approach in drug development, first developed by Dr. Joseph M. Juran. And then it starts with predefined objectives, which emphasize on a clear understanding of the process as well as the product itself. So then with these objectives, we then use them to identify the product and the quality attributes of the product to be used in the implementation of the quality by design process. So before the implementation of quality by design in the pharmaceutical industry, the quality of a product was only assured by end product testing. So now with QBD, we can ensure the quality of the product through its formulation and manufacturing process. And then moving on to the motivation for our study. So administering oral dosage forms for pediatric patients, especially ones who are neurodivergent, can present challenges, especially for caregivers and healthcare professionals. So these challenges can ultimately cause a decline, one, in patient compliance and affect negatively the therapeutic outcomes. So then we aimed to formulate paracetamol chewable tablets using QBD to cater for the different needs of patients and to give healthcare professionals and caregivers a larger variety of options to choose from when administering medication. Our research question: what are the quality attributes of formulating paracetamol chewable tablets using quality by design? And then our aim, we aimed to identify and optimize the quality attributes required for the formulation of paracetamol chewable tablets. We move on to our objectives. So then, our objectives: one, was to synthesize paracetamol active ingredients. Two, to analyze the properties of paracetamol using specifications according to pharmacopoeia. Three, formulate paracetamol chewable tablets using quality by design approach. The last one was to evaluate the project's quality control tests, such as hardness, disintegration, and weight uniformity. Our methodology was divided into two phases. Phase one, which was the synthesis of our API, and phase two, which was the formulation of our product. So it is aligned with our objectives. Here we have a summary for you of the methodology of our research, whereby step one was the synthesis of our API. Step two, we analyzed the API properties. Step three, we formulated paracetamol chewable tablets using quality by design. And then the last step was the evaluation of the quality control tests. And then I'm moving on and giving to my partner.
Okay, so our study consisted of two phases. So the phase one was the synthesis of paracetamol, which is our active ingredient. So this slide only shows the raw material that we used. So paracetamol was synthesized using diazotization method, where acetanilide was treated with nitrous acid. Ultimately, it was vacuum filtered, which eventually formed the solid particles that were formed there, which is our final product, which is the active ingredient. So after we synthesized the active ingredient, we analyzed it, where we conducted the absorbance test, where at the volume of 6 mils, we found that the absorbance of the active ingredient that we synthesized was the same as the one that was brought to us, which is the commercially prepared paracetamol. So after that, we made the percentage yield, which was found to be 13.5%. Which means most of our product was lost, because this is a relatively low number. It could be due to the poor filtration technique that were used in our synthesis of the API. Also, the percentage purity was found to be 95%, which means 95% of our product was pure, with the remaining approximately 5% consisting of impurities. We also made the melting point according to pharmacopoeia standard. The first two of our formulation consisted of formulating our chewable tablets using quality by design. So this slide only shows the list of ingredients that were used. So in total, we had 3 formulations, same ingredients, same methods. The only difference between the formulation is the varying quantities of some excipients that were used. So this flow diagram shows the methods that were followed, where we were formulating the paracetamol chewable tablets by melt granulation. As you can see in the picture, after all the methods were followed, we poured the final product into the non-sticky mold, which was left for 2 to 3 days for the tablets to form. So after the formulation of the paracetamol chewable tablets went to the results and discussion. So we incorporated quality by design into our research by establishing quality target product profile, which is the summary of quality characteristics that ideally should be achieved to ensure the desired product. So you can see, among others, there's a taste which should be palatable, because this formulation is meant for pediatric, so the taste should be palatable to ensure acceptance and compliance. Also, the dosage strength is supposed to be 125 milligrams. So again, this is the continuation. Our tablet was pink in color and cube. Okay.
So from our QTPPs, we found our critical quality attributes. So what are those? These are physical, chemical, biological, and microbiological characteristics, which when they are not in specification, they have a direct impact on our efficacy and our safety of our product. So from the FDA guidelines to chewable tablets formulation, in 2018, we found or we identified our taste to be part of our CQA, and then our disintegration and tablet hardness were also identified as part of our CQA. However, weight and friability were not identified as part of the CQA. They were also measured throughout the process to ensure the quality of our product was consistent, which, by the way, is the intent of quality by design. So we also performed the risk assessment for our product, which is just a systemic process that identifies and manages the possible risk that our product might have. So critical material attributes, these are properties of the raw materials that have a direct impact in our product. So you can see from the table that we have acacia and raspberry syrup that had a high impact on our hardness. So the risk assessment that was used was according to the GAM chart, which is the Good Automated Manufacturing Practice Guidelines. And from here we can see that we had low, medium, and high risk according to the chart. And then this is just a summary of what was in the previous slides. Paracetamol affects the taste, and then starch is, as a binder, affects the hardness, which means it's also part of our high risk. And then critical process parameters, these are key variable or conditions which must be kept in specification so that our product remains consistent in terms of quality and safety or efficacy. So we can see here that cooling time, temperature, and mixing parameters have a direct impact or a high impact on our hardness, disintegration, and weight uniformity, respectively. So as my partner has already alluded before, these are the formulations that we used with varying quantities of raspberry syrup and total water content, which affected our results. So on to the results then. Weight uniformity, we can see as displayed on the slides that there are some discrepancies with our weight uniformity, which was slightly due to poor transfer techniques, since we had to pour the mixture into the non-sticky molds, and could also be due to the raspberry syrup and total water content used, which affected the thickness and of the product. And then the disintegration, formulation 3 had the fastest disintegration, which is ideal for pediatrics. And then for hardness, formulation 3 had a failure result. This does not mean it was a failure in terms that the product was too soft for the machine to measure the results. However, it was not soft like a powder tablet that it crumbled under pressure, but it was soft like a gum. You can say it was squishy like, so the machine was not able to measure the hardness of the tablet. And then friability, all our formulations failed the friability test, since the friability was supposed to be less than 1%. And then the testing was done voluntarily by all group members. Formulation 1 had a bitter taste, formulation 2 had a bitter aftertaste, and then formulation 3 was palatable with a raspberry taste in it. So as part of our control strategy, we deduced that formulation 3 was the ideal formulation, with the raspberry concentration and total water content being 8 mils and 15 mils respectively, and this can be scaled up to the required quantity if needed.
So in conclusion, On the first phase, we successfully synthesized paracetamol according to the diazotization process, and we found that the melting point is 168 degrees Celsius, percentage yields 13.54, while the percentage purity was 95.37%. So on the second phase, 3 formulations of paracetamol tablet were formed by melt granulation method using quality by design. After evaluation of the test, we found that the most stable formulation is formulation 3 because it was soft, palatable, and had faster disintegration time. So from the start, we were able to identify and optimize critical quality attributes for the synthesis and formulation of paracetamol chewable tablets. So recommendation, researchers should conduct dissolution studies to determine how well the API is released from the chewed particles. Studies should be considered as a CMA, perform a design of experiment for future studies. These are our references and
any questions for the paracetamol? Ooh, that corner is dangerous. That corner is dangerous.
So we often in pharmacy get the new products that are brought on the market, everything is sugar-free. So did you at all look at another flavoring that could have been used in place of the raspberry syrup for a diabetic patient?
So for a diabetic patient, we also check that they are both the syrup and the concentration. So we found that the concentrated syrup can be used for a diabetic patient. Yeah, the syrup and juice. Yeah, the juice one.
Next question, mine's in 2 parts. Why did you want to firstly synthesize your active material, seeing as though your yield was so very, very low from that? And we know normally our raw materials we just buy in. So why was that a decision? And then secondly, why was assay not a critical quality attribute? So why the choice to synthesize rather than buying in your active pharmaceutical ingredient, which is the norm? And the second part was, what was the rationale for assay not being a critical quality attribute?
Okay. So we, as part of our research project, our phase 1 entailed that we had to synthesize our product. However, the product that we used for formulation was the commercial one. So we needed to synthesize so that we can see how the API properties are before we can law. Then in terms of the assay, it was not part of the CQA because of the necessary resources to measure those things, the assay.
One more question, Prof.
Thank you for your presentation. I just want to find out, so palatability can be quite subjective. Did you guys only test that amongst yourselves in your group? Or for example, did you hand out some of the products to your classmates to assess, just to find out about the taste? Thank you.
Unfortunately, Prof, we were the only willing test subjects at that time. So it was only between the group members.
Okay, I won't allow more questions because I'm having network problems, so I'm trying to kill the time. I'm seriously having network problems. so you can continue with the question.
Thank you so much. I think for my question will be a bit lighter because I'm not a pharmacy person, and it's just out of curiosity. There is a slide where I can't remember but all the tests failed, and I was just wondering, do you have any assumption as to what led to that, and what are the implications of those tests failing? You can look at this.
Okay, so the test that failed, it was the friability. So in terms of the friability, it varies and depends largely on the parameters that we used. So the parameters we used was rotations, 25 rotations per minute for 4 minutes. So in different conditions, it has different implications and different results. And I forgot what was your second part of the question.
The second part was like, what is the implication for the failure? But you have highlighted that. Thank you.
And also just to add on, since friability isn't a critical quality attribute for our chewable paracetamol tablets, it wouldn't affect the safety and efficacy of the tablets themselves.
Okay, there's Prof.
Thank you. Thank you very much for an interesting presentation. If we just look aside of the assay and the failures, I had 3 or 4 questions, as you were presenting. But as you got to the end of your presentation, you seem to have answered those questions. And I want to compliment you in the way you handled this project. I think it was an excellent project. And I'm glad you didn't, no disregard for Prof. Boydenstein, but I'm glad you didn't test it on the class, because that would have raised a huge ethical problem. But good luck. Good luck with your future quality assurance officer careers too.
Ladies and gentlemen, our honorable judges and my colleagues here present, I'm Hopo Moyo, and with my colleague Casey Els presenting on the research topic, investigating the most reported household agents for off-label medicinal purposes. This is the structure of our presentation. Thank you.
Masks are a thing of the past. However, COVID-19 has played a vital role in highlighting the off-label medicinal uses of household agents, with the WHO estimating that between 65 to 80% of the world's population uses complementary and traditional medicine. In retrospect, the use of these agents in remedies such as Boereeraad and indigenous African knowledge were one of the earliest forms of treatment available and remain pertinent centuries later.
Complementary and alternative medication, as mentioned by my colleague, has been defined by the World Health Organization as a broad set of practices that is not necessarily part of that country's traditional or conventional medication and is not fully integrated into the healthcare systems of that particular country. While this conventional medication and complementary and alternative medication are 2 separate ideologies, there's a constant overlap of a goal of achieving health, which is observed in both practices. Household agents such as ginger, turmeric, and honey have been widely used for their nutritional purposes. However, social media has reported the use of these agents in playing a critical role in disease and ailment treatment, prevention, and alleviation. And thus, the purpose of this research is to scientifically assess the rationale behind the use of these agents for the reported medicinal purposes. Thank you.
Whilst these agents have buffered against the financial and medical destitution burdens associated with conventional therapy, the use thereof often occurs in the absence of scientific knowledge and risk-based analysis pertaining to the pharmacological action thereof.
With that problem being identified, that brings us to the aim of our study, which is to explore the reported household agents commonly used off-label for medicinal purposes and the scientific reasons that underline their use. To achieve this aim, our objectives are to utilize publicly available data as a primary source of information and to use and evaluate scientific literature that identifies the proposed pharmacological mechanisms behind the use of these agents, and lastly, to draw conclusions and hopefully make recommendations for future. Therefore, this study was conducted as a systematized review, and a new Gmail account was created to avoid the risk of bias in logging into the different social media platforms. With that Gmail account being created, search prompts were used such as South African traditional medicine, Burarat in South Africa, household agents for off-label medicinal use in South Africa, and these search prompts were used for 4 different social media platforms, that is Instagram, TikTok, blogs, and Facebook. The top 10 most appearing searches on the social media platforms were then identified, and these were organized in order of appearance. After comparing these various pros from the 4 social media platforms, the top 3 household agents, which is ginger, turmeric, and honey, as identified by our statistical analysis, were then compared to relevant scientific literature, which falls within the inclusion criteria of our study, which is English language and scientific literature that has been peer-reviewed and that appears in PubMed Central, Science Direct, and Google Scholar as per our NMU database. These results were then reported in form of a tabular accompaniment as per the systematized review of study, and therefore the results that we're presenting to you now are the results that were obtained from the searches that were done. Thank you.
Honey, whilst being a delectable treat found in the pantries of most South African homes, has also been reported for the off-label use in both tonsillitis and sore throat. With emphasis on the type of honey, it is proposed that pure honey contains glucose oxidase, which is metabolized into hydrogen peroxide and subsequently results in bactericidal effects against commonly implicating organisms, such as Pseudomonas. Furthermore, honey is also known to inhibit both cyclooxygenase 1 and cyclooxygenase 2, thereby reducing inflammation by inhibiting the inflammatory cascade and directly inhibiting the formation of inflammatory mediators such as prostaglandin E2. The previous benefits of honey described, in addition to honey's ability to penetrate the epidermis, antifungal properties, and the ability to reduce melanin production, also supports the potential use of honey in atopic dermatitis, commonly known as eczema, hyperpigmentation, open comedones, wounds, burns, and dysmenorrhea. The hygroscopic properties of honey, which ultimately dehydrate bacteria and lead to cell death, combined with the hypothesis that the sweetness of honey promotes saliva secretion, which liquefies mucus and directly stimulates nerve endings for antitussive properties, further supports honey's potential use in coughs. Interestingly, while a risk-based analysis of honey showed that honey had minimal side effects, the use thereof is not recommended in children under the age of 1 year old due to the risk of infantile botulism syndrome. Additionally, in weight reduction, honey in animal studies have suggested that the use of acacia honey is superior to that of conventional therapy such as oralistat in weight reduction in mice modules. The proposed mechanism is related to prolongation of gastric emptying time, which reduces appetite, promotion of lipolysis, that is the breakdown of lipids, and inhibition of lipogenesis, the formation of lipids.
Ginger, a commonly used herb in the world, contains 2 phytochemicals. First, which is volatile oils, and these are responsible for the fruity smell of honey. Non-volatile oils, these are responsible for the pharmacological mechanisms that are associated with honey, and these contain constituents such as shogals, gingerons, and paradols. Our Out of these constituents, 6-gingerol is the most common, and these differ in structure due to the number of alkyl chains that are contained in these different substituents of ginger. Ginger has been used to manage bronchitis, obesity and overweight, tonsillitis, pain and inflammation, as well as colds and flu. Some of these mechanisms overlap with those previously described in honey and those that are going to be explained in turmeric. Ginger has also been found to work against nausea and vomiting, and this occurs mainly by the antagonism of serotonin 3 and muscarinic 3 receptors. This mechanism ultimately results in an inhibition of afferent input to the central nervous system, which is stimulated by the neurotransmitters that are released in the gastrointestinal tract. Furthermore, ginger has been shown to stimulate oral and gastric secretions, as well as regulation of gastric motility, which provides a control in nausea and vomiting.
The benefits of turmeric, also known as the golden spice, in addition to the previously explained properties in both ginger and honey, have also seen it potentially being effective in digestive ailments, acne, hyperpigmentation, weight loss, and tonsillitis. Their mechanism of action may be attributed to similar pharmacological action of anti-inflammatory and antibacterial properties. The benefits of turmeric in influenza have been identified as antiviral properties against commonly implicating organisms such as the Epstein-Barr virus, and anti-inflammatory and antioxidant properties. In vitro studies have proven the safety and efficacy of 50 milligrams of turmeric dosed 3 times a day. However, further clinical research is required to confirm these results in human subjects. Similarly, the use of 500 milligram turmeric daily in osteoarthritis has been known to inhibit the degeneration of chondrocytes and restore both intrinsic type 2 collagen and glycosaminoglycan. In hypertension, the mechanism of turmeric is not well understood. However, it is proposed that turmeric stimulates the production of nitric oxide synthase, which ultimately restores glutathione and is known to also improve the antioxidant capacity of cells. Further mechanisms proposed include the increasing of nitric oxide levels, as well as the stimulation of the NRF2 signaling pathway, which potentiates the effects of acetylcholine and results in vasodilation, ultimately reducing the peripheral vascular resistance. Taking into consideration that this study did not employ animal or human subjects, ethical clearance was not considered. The potential limitations identified in this study were identified as time constraints, limited access to data, and potential misinformation from social media platforms.
In conclusion, ginger, turmeric, and honey have been shown to have pharmacological properties that can work against the alleviation of different ailments. And there is need for further research for the dosages and the dosing frequencies, and to educate healthcare professionals on the safety and efficacy of these agents. Lastly, we must listen less to the opinions of those who either overtly promote or stubbornly reject the use of household agents without supporting evidence. The many patients who use this complementary medication deserve better, and therefore its use should and hopefully will be determined by unbiased scientific evidence. We would like to thank our supervisors, Dr. Luke Mekele and Dr.
Thank you. We'll now open the floor for questions.
And to think that they wanted today to be a day off because of Ramaphosa. Look at how beautiful they are. Any questions?
Firstly, have you seen the price of honey, ginger, and turmeric lately? But aside from that, the use of turmeric for acne, is that topical or is it oral?
Okay, so the use that we saw was topical, and oftentimes a lot of the research was proving that the use of turmeric in combination with pure honey, specifically acacia honey, applied to the skin, but then that came with limitations as well, because turmeric is known to stain the skin. And so that was one of the things that we took into consideration and that we did include into our treatise, and that ultimately led us to our conclusion that so many times, because we don't have the scientific literature available and we don't have enough healthcare practitioners educating patients on complementary and alternative medicines, patients are somewhat forced to use social media platforms for their information. And this is where they aren't properly educated on both the risks and benefits of using these treatments in combination or in monotherapy. Thank you, Ms. Worthington. Thank you.
Sorry, I know there was another question there. You spoke about you took 10 items or 10 of the most common household agents, but you only discussed 3. What were some of the other agents that came up quite commonly on social media?
Okay, thank you very much. So in terms of 10, it was the top 10 posts appearing on social media. And then, so we collated that. So we had Facebook, top 10 on each search prompt. So we had South African traditional medicine as a search prompt that produced 10 results. I mean, more than 10, but we only took the first 10. And then another search prompt, Burarad in South Africa, we took the first 10, and then we collated all of that, and then we put it together, and then we identified the most 3. We would have loved to do more, but because of time constraints, we could not. So some of the agents appearing were lemon, things like cinnamon, and also those are actually used in combination with the agents that we have described also today. Yeah, thank you so much.
One more question and then...
I just want you to just, in terms of honey or the 3 products that you focused on, I realized that you, on your slides, you had the articles or scientific literature that supported some of the benefits, and I would love just for you to reflect on some of aspects that were reported on social media but were not supported by the literature that you found.
Thank you for your question. In most cases, we were able to find literature, however, with varying degrees of evidence. So some of the literature that we found, for example, in honey, it was not supported in the use of stretch marks, and that we explained in our research. But in cases where the articles were against the use of honey in these agents, so for example, honey in coughs, we had a look at conflicts of interest and the limitations of the studies. And oftentimes there are conflicts of interest with pharmaceutical companies sponsoring those studies. But in most cases, we were able to find literature, and even in, as I said, weight loss, which was quite a surprise to me that honey can be used in weight reduction as well.
Okay, thank you very much. So now we're moving back to the lab, to the preclinical studies now. We're going to look at the antiretrovirals, and we're going to look at anti-cancer. So starting with the antiretroviral group, I see their supervisor is here, so they can come forward and start preparing their presentation. But I must say, when during COVID, there was a page where people would share remedies, with other people. The one most common remedy that was shared there was onions and lemon. So I'm not sure exactly if the previous group came across, but the onions, they took the cup, the onions. Everyone, they even put onions in the socks, inside the socks. They said it helped with COVID. So that was quite an interesting one. So antiretroviral group. Now we're moving back to the laboratory. Come guys. More testosterone. There is your pointer. What? Okay, starting now.
Okay. Good afternoon, fellow colleagues and lecturers. My name is Munashe Mkomba Choto, and I'll be presenting our research, which is entitled antiretroviral therapy, and risk of adverse pregnancy outcomes, particularly the impact of combined antiretroviral therapy on trophoblast cell proliferation and migration. South Africa is the most HIV-affected country, where 7.5 million people are HIV positive, and 4.8 million people of those are women of reproductive age that is greater than or equal to 15 years of age. Therefore, combined antiretroviral therapy is now being used during pregnancy to prevent mother-to-child transmissions, as it was seen to reduce the rates at 6 weeks postpartum to below 3%. However, due to the national burden of the disease, South Africa also adopted the WHO guidelines that recommend all HIV-infected pregnant and breastfeeding women to commence and continue lifelong antiretroviral therapy, irrespective of the clinical or immune status. According to the South African guidelines, the first-line antiretroviral therapy for pregnant women comprises tenofovir, lamivudine, and dolutegravir. However, it was seen that the use of combined antiretroviral therapy during pregnancy can lead to adverse pregnancy outcomes, such as preeclampsia, stillbirth, preterm delivery, small for gestational age births, and lower birth weight. And these conditions has also been associated with increasing the risk of infant morbidity and mortality, especially in low-resource settings. A recent published paper from a facility-based cohort in Botswana also found out that HIV-infected women that were either on antiretroviral therapy or using short-course preventative mother-to-child regimens were at an increased risk of adverse pregnancy outcomes. And these associations appears to be due to maternal antiretroviral therapy, which was also found with an up to 1.8-fold increase in adverse pregnancy outcomes. There have been several mechanisms that have been proposed to account for this association between the combined antiretroviral therapy and adverse pregnancy outcomes, some of which include the link between placental insufficiency and combined antiretroviral therapy-induced adverse pregnancy outcomes. This slide shows a picture of the placenta, particularly the placental formation. And as we know, the placenta is important in pregnancy as it facilitates the exchange of nutrients as well as gases between the mother and the fetus. So the placenta is formed in the first trimester. And in the first trimester, extravillous trophoblast cells invade the uterine wall. And this invasion will cause transformation or remodeling of the maternal spiral arteries. And these maternal spiral arteries are transformed from high resistance, low capacity vessels to low resistance, high capacity vessels that are able to facilitate pregnancy as well as to make sure that pregnancy is safe. So therefore, the impairment of this transformation or remodeling is what will lead to placental insufficiency, therefore causing the adverse pregnancy outcomes. Which therefore led us to the research question that stated that does combined antiretroviral therapy drugs have an impact on trophoblast cell proliferation and migration? And we aimed to understand the mechanisms by which combined antiretroviral therapy drugs may be implicated in pregnancy-related adverse conditions by examining the role of combined antiretroviral therapy on trophoblast cell viability, proliferation, and migration. We hypothesized that combined antiretroviral therapy may contribute to placental insufficiency via the inhibition of trophoblast proliferation, resulting in adverse birth outcomes reported with ART use. Therefore, our objective stated, we wanted to investigate the effects of the individual and combined antiretroviral therapy drugs on trophoblast cell proliferation in HTR8S venereal cells, as well as to investigate the effects of the combined antiretroviral therapy drugs on trophoblast migration in HTR8S venereal cells. As for our methodology, HTR8S venereal cells were subjected or were treated with varying concentrations of the tenofovir, lamivudine, and dolutegravir, as well as the fixed dose combination TLD for 24, 48, 72, and 96 hours. They were then subjected for a biochemical assay for profile analysis, where cell viability was determined using trypan blue. Cell proliferation was determined using MTT assay and validated with CFD assay. And lastly, cell migration was determined using the Scratch migration assay. And then our results were analyzed using Graphite Prism version 10, where the data was presented as a means of positive or negative standard deviations, and the comparisons and statistical significances were determined using two-way analysis of variance, where the p-value of less than or equal to 0.05 was considered statistically significant, and all experiments were done in triplicates. Our results show that cells that were treated with individual components of TLD, as well as the fixed dose combination TLD, had a significantly reduced number of viable cells compared to the control at all time points. And this significant reduction was more profound at the highest concentrations. A similar trend was also observed with the MTT assay, which was validated with the CFD assay, where individual components of TLD as well as the TLD fixed dose combination significantly reduced cell proliferation compared to the control at all time points. And again, significant reduction was more profound at the highest concentrations. Due to the significant inhibition that we noted after conducting the cell viability and proliferation results analysis, we observed that fixed dose combination TLD at the highest concentration had increased inhibition compared to the individual antiretroviral therapy drugs. We therefore conducted a scratch migration assay to further observe these effects of fixed dose combination TLD at the highest concentration, where we also saw that the cells that were treated with fixed dose TLD had a significant inhibition of cell migration compared to the control over 48 hours. So in overall, our results indicated that cells that were treated with individual components of TLD as well as fixed dose combination TLD showed a significant reduction in cell proliferation, viability, as well as migration in the first 24 hours, suggesting that treatment beyond 24 hours had detrimental effects on placentation, as it inhibited trophoblast proliferation and its invasion into the maternal decidua, thus leading to placental insufficiency, which then causes the adverse pregnancy outcomes. We therefore conclude that first-line combined antiretroviral therapy drugs inhibited trophoblast cell proliferation and migration, and may therefore contribute to adverse pregnancy outcomes associated with combined antiretroviral therapy used in HIV-positive patients. Future studies should examine the effects of combined antiretroviral therapy drugs on trophoblast cell cycle progression, and to also determine in which phase of the cell cycle the cells are arrested due to the ARV drugs. We would like to acknowledge our supervisor, Dr. S. Nzuza, as well as our co-supervisor, Professor J. Bodenstein, for guiding us and helping us make this research study a success. Ms. J. Van Yarsvoort for helping us procure all the resources that were required so that we can be able to conduct the experiment, as well as the Human Physiology Department for helping us with our lab work. This marks the end of our presentation.
Thank you, guys. Any questions for this group? They're all looking beautiful. Their white jackets.
Thank you very much for your presentation. My question relates to the big picture that you have painted in your background and introduction with regards to the use of ARVs. I'm particularly more interested at the statistics that you may have come across with regards to stillbirth in South Africa, and perhaps what could be done then going forward with improving the safety of these products, or what did you come up with in terms of your review of literature, in terms of what can be done going forward to prevent such adverse effects?
Okay, thank you very much for your question. So according to the literature, we found out that most literatures were actually in support of using ARTs like tenofovir, lamivudine, and dolutegravir, right? Basing on the fact that they were saying that it's safer, but not like completely stating that like the adverse effects that come with it, right? So, but there were only a few articles that were actually stating that no, there's actually a risk of having those adverse pregnancy outcomes. So we wanted to verify, to validate that is there an actual, because right now we are saying in pregnancy, because back then we used to say you have to give TOD maybe after 6 weeks. Right, but now we are saying that even when you are beginning, when the pregnancy is beginning, you just have to initiate TOD because it's the safest of all the ARVs, right? So in terms of risk benefit analysis, we are saying that is for now that's what we have at the moment to say TOD is safer, but there are adverse pregnancy outcomes that are there. So we are recommending that there be more research into developing maybe safer and newer ARVs that actually exclude the possibility of adverse pregnancy outcomes.
Any questions? Any more questions? I'm sure they're smiling quietly next to me. Okay, guys, thank you so much. Okay, are you guys ready? Yes. Starting now.
Greetings, everyone. My name is Lucanio, and these are my colleagues. So we were doing a pharmacodynamic study that involved the screening of natural antioxidant against cancer activity. So this is our table of content. So according to the Cancer Foundation, cancer is the leading cause of death globally with 9.6 million deaths per year. And our study focused more on the melanoma cancer, which is ranked the 5th to 6th prevalent cancer in South African men and women. Cancer is caused by uncontrolled cell growth in the body, and this abnormal cell growth can form benign tumors as well as malignant tumors through a cell cycle, which is the process by which normal cells and cancerous cells, they divide and produce new cells. Cell cycle has 4 stages: gap 1 phase, the S phase, gap 2 phase, and the M phase. There are checkpoints that help prevent cell cycle by spinning out of control. But when these checkpoints have been disturbed, it can lead to malignant cells. Malignant tumors have a potential to infiltrate and metastasize nearby tissues. And one of the hallmark features of tumor cells is their ability to evade apoptosis, which is a programmed cell death. So there are intrinsic and extrinsic pathways that contribute to apoptosis, and our study was more focused on the intrinsic, which included oxidative stress amongst others. Oxidative stress is an imbalance between free radicals as well as the antioxidant enzymes. And these free radicals are essential physiological function at low concentrations. So basically, this is what is supposed for normal cells. There should be a balance between the antioxidants as well as the free radicals. But when now there's oxidative stress, which is that imbalance, the long-term oxidative stress damages the DNA, the proteins, as well as the cells, promoting carcinogenesis as well as tumor progression. So, since our study now is researching the antioxidants, antioxidants are able to minimize the oxidative stress by donating electrons, which balance the free radicals and reduce this reactivity. The natural oxidants that we used in this study include uthymol, that is found in red wine, curcumin, which is found in turmeric, as well as thymol, that is found in essential oils of thyme. The aim of the study was to determine whether natural antioxidants, namely thymol, curcumin, and resveratrol, have selective anti-cancer potential in the melanoma cells. Our objectives included to which is objective 1, we wanted to determine the antioxidant activity of the compounds using the OREC assay, to determine the cytotoxicity of the compounds on the HS27 fibroblast, which are normal cells, and melanoma cells, which is the cancerous cells, using the host and propidium iodide. Objective 3 was to determine the ability of the compounds to resist oxidative stress in the normal and the cancerous cells using cell rocks orange. Then objective 4, we wanted to perform a cell cycle analysis that was going to help us to establish whether the compounds, they targeted the apoptotic pathway and the cell cycle arrest. In our methodology, where we wanted to identify the antioxidant activity, we used OREC assay to screen the antioxidant capacity of the 3 compounds. And this study was a kinetic study where we measured the fluorescence at different time intervals. Trolox, a synthetic vitamin E with known antioxidant capacity, was used as a control. And to determine the antioxidant capacity of these compounds, the net area under the curve for each sample compound was calculated, and the Trolox equivalent was calculated from comparison to a Trolox calibration curve. The HS27 and the MEWA cells were grown in a DMEM low glucose medium supplemented with 10% FBS under aseptic conditions. These cells were then seeded using the sterile 96 well microtiter plate at 5,000 cells per well. And they were left overnight to attach in a 37-degree incubator with 5% carbon dioxide. The various dilutions of the treatments, which included thymol, curcumin, and resveratrol, as well as cisplatin, which is a known antioxidant that was used as a control, were prepared in a complete medium. And the cells were then treated with the dilutions and were left in the incubator for 24 hours. So, okay. To indicate the apoptosis, during apoptosis, which is the programmed cell death, the membrane phospholipid, known as phosphatidylserine translocation, is translocated from the inner to the outer leaflet of the membrane, allowing a dye known as annexin V to bind. Under normal physiological conditions, the level of reactive oxygen species in cells are controlled by the balance between the antioxidant and the free radicals. Excessive production of the ROS can induce apoptosis. The images of the treated cells were acquired and analyzed using the Image Express. The cytotoxicity of this HS27 and the MEWA cells was determined using the Husch and the propidium iodide dyes. So looking at the table, we have the Husch dye, which binds to the DNA because of its membrane permeable, and it then stains the nucleic acids. And these are the wavelengths at which it's excited, as well as it emits the blue fluorescence. Then we have propidium iodide, which is membrane impermeable, so it is only able to bind to non-viable cells. We also have cell rocks orange, which is a cell permeant dye that binds to the reactive oxygen species, indicating the oxidative stress. We also have annexin V, which was illustrated from our previous slide, that is able to bind to the reactive oxygen species, to the phospholipid. So, in the results and discussion, we found that in the OREC assay, These were the standard curves that were obtained by plotting the net area under the curve of the different compounds against their concentration. And each curve was subjected to linear regression analysis for comparison to a Trollocs standard curve. We then calculated the Trollocs equivalent from the ratio of the slope of the linear regression of the compound to that of Trollocs. And we found that resveratrol had the lowest Trollocs equivalent value, which was then followed by curcumin, then lastly thymol. And we concluded that resveratrol showed higher antioxidant capacity compared to the two compounds. On the cell rock study, curcumin resulted in an increase in oxidative stress that was dose dependent in Mewar cells. And this is indicated by the 3 stars on top of that. So as we can see that as the concentration increases, there's also an increase in the oxidative stress that is caused by that curcumin. With resveratrol, it inhibited the oxidative stress in both normal as well as cancerous cells. Thymol in cancerous cells resulted in an increase in oxidative stress, which was more significant at concentration 100 micromolar. Results of cell cycle analysis. In the cell cycle analysis, curcumin at concentration range of 1,25 to 10 caused S-phase arrest in the cancerous as well as the normal cells. And as the concentration of curcumin increased, there was an increase in apoptosis on both the normal as well as the cancerous cells, which was dose dependent. Resveratrol at concentration 3,125 and 50 resulted in S-phase arrest in both cell lines. Thymol also resulted in S-phase arrest in the Mewar phase at concentration 6,25 and 100 micromolar. There was an increase in the early apoptosis and the late apoptotic cells of the Mewar cells and the HS27 that was caused by curcumin, which was dose dependent, and we then came to a conclusion to say that curcumin appeared not to be selective in this way, as it caused cell death in both the normal as well as the cancerous cells. Thymol also resulted in a slight dose dependent increase in early apoptotic cells of the normal as well as, but there was no significant results on the cancerous cells. And there was a dose-dependent increase in the early apoptotic cells in both the normal and the cancerous cells. And resveratrol also showed no selectivity as it affected both cell lines. We then concluded to say that resveratrol has an anti-cancer potential as it had the highest antioxidant capacity in the OREC assay. Curcumin and thymol also showed anti-cancer potential as they increased the oxidative stress in cell rocks study in the Mewar cells. All 3 compounds resulted in the cell cycle arrest at S-phase in the cancer cells, and this is important in cancer cells as it stops cells from replicating. Curcumin induced apoptosis in cell cycle analysis in both cell lines, which was non-selective. Curcumin also caused early apoptosis in both the cancer cells and the normal cells. Further research needs to be done, and future experiments that can be done include the caspase 3 and the caspase 8 activation, as well as histone phosphorylation, etc. We want to acknowledge the MNRG group as well as the National Research Foundation, as they allowed us to use their environment as well as their equipment. Also, special thanks to our supervisors, Dr. Dambuza and Ms. Kritzinger, and yes, our lady there, Usis Nopasika. These are our references, and thank you.
Nopasika, shine, Nopasika. I wanted to learn how to work in the cell lab, so. Any questions? Any questions?
Thank you. Thank you. It seemed like a very complex project, but I think all these biochemistry projects are quite complex and sensitive. I just wanted to know, what is your opinion of curcumin? Because it seems to have both good and bad properties over here. And as far as I recall, I may not be 100% correct, is it not an ingredient of, or a constituent of turmeric?
Yes. Oh, yeah, we did indicate in our presentation that it originates from turmeric. And from the results that have been shown here, it has been shown not to be selective, as it would induce apoptosis, which is the cell death in both cell lines, which is the normal cells as well as the cancer cells. So I feel like more research needs to be done in that, and also focusing on the cell cycle. We may also need to be able to, okay, let me not go overboard.
Thank you for that very complicated presentation. You did very well navigating it. My question is, I don't know if I should ask, did you use the same quantities or the same concentrations for the 3 products that you used as antioxidants in terms of the minimum effective concentration as an antioxidant?
Okay. Good afternoon, everyone. So the concentrations that we used, they varied, like they were different for each compound, and they were based on literature review. So that's where they were taken, and then they were experimented as per each compound.
Any questions? One more question. There's a question over there at the top.
Hi. Afternoon, sorry. Are you able to hear me clearly?
Yes.
Okay. So any type of laboratory study, challenges can occur, problems happen. So what were some of the challenges that you experienced, and how did you mitigate those challenges?
So working with the cells and having to grow them under aseptic conditions, it was a bit challenging, as we would have contamination. So I think that was one of the challenges that we had, contamination of the cells. Then we had to go back to the drawing board and grow another cells again. But we did have cells that were healthy, that we would ensure that, yeah, those ones no go. We we keep them. Yeah. So yeah.
That was the ZRE group. These are the full projects that were done throughout the year. And as you can see, they were very interesting. We had quite a huge variety. We were in the pharmaceutics, and then we were in the household contents, and all the home remedies that we try and use at home. And then we went on and we did preclinical in the laboratory. As you can see, both groups worked with cells. It's not easy to grow cells, as they said, that you get contamination and you want to blame someone. It's just one of those huge setbacks. So our students, and I'm sure Maxine, you will agree here, they've learned a little bit of resilience, they've learned a little bit of grit in this journey, and they've got a little bit of glimpse of what research is about. It's not a smooth sailing. Sometimes you want to pull your hair because you're not getting what you want, but you develop a very strong character, and also adjusting outside your comfort zone. So now we're going to break a little bit, have a little bit of nibbles outside, and then we're going to come back. We are a few minutes late, so let's make it 3:30. We come back, and then we're going to start with the posters. The posters now are going to be short and quick because they've already projected outside. So these were the top posters that were judged, and so they're going to just tell us what they did very quickly, in and out, so that we can wrap up the program.
Good afternoon, everyone. We are Group 1 from the game-based learning elective. Our our project was the development of a game-based learning tool for the revision and improvement of cardiovascular care, and we created a group. Game-based learning is an active learning technique used to educate through game experiences to improve students' critical thinking. Gamification is the incorporation of game elements into traditional activities such as point and reward systems. The use of game boards in game-based learning has shown to build teamwork, communication, and problem-solving skills in students. Studies have found that game-based learning shown to be effective when used to supplement studying, since students found it more pleasurable and stress-free. In South Africa, South Africa is experiencing an increase in cardiovascular disease burden, and it's the second highest cause of death. Cardiovascular disease treatment approaches are complex, and the pharmacology is quite extensive, as we all know, and it can be very stressful and overwhelming. Therefore, we created and designed a fun game which helps students in the supplementation of their studies. We are aiming to develop a new game that will help supplement student learning to better understand cardiovascular care in the BPharm program. Our objectives were to design a game to improve comprehension of cardiovascular drugs and create a game that clarifies the understanding of cardiovascular drug use. We utilized the mechanics, dynamics, and aesthetics framework. Mechanics are the tangible parts of a game, like the game pieces such as the board, the dice, our cards, and a timer, and how the game pieces are utilized. The dynamics is the process of play, like the time pressure created from a timer, and problem-solving skills when presented with a challenge. The aesthetics are the things that players feel, and these feelings can be categorized according to sensation, fantasy, narrative, challenge, fellowship, discovery, submission, and expression, and these vary depending on the type of game that we play. The results. Our objectives have been met through the implementation of the MDA framework in our group of 3. We designed and created a game board targeted at completing our aims. The implementation of the mechanics like cards, dice, game pieces, and a timer to aid in the dynamic of critical thinking and time pressure and rules. To allow the achievement of the fellowship, the achievement of our aesthetics, and in our game, these were challenge, satisfaction, and fellowship. We reached the decision to make cardiovascular pharmacology the theme of our game by using our past experience with studying the topic, everyone can agree, and researching cardiovascular disease burden in South Africa, because it's very important for us as pharmacists. After finalizing on a theme, we focused on first deciding the type of game we were going to create, which was a board game. And then after that, we discussed the type of mechanics we wanted to utilize and the aesthetics we were aiming to achieve. We then completed this by picking a theme, using a color, font, and graphics to match that theme. We then completed a rough draft for our supervisors, and they guided us on fixing or changing aspects. Overall, the designing of the game required a lot of effort, and it was a fun experience and helped us even effectively review out this topic. Our objectives of the study has been met through a board game and the aims of supplementing and aiding our study of cardiovascular drug therapy in South Africa. Due to ethical constraints, the effectiveness of our game unfortunately cannot be tested. However, theoretical constructs of the game is hopeful, and in the future, with ethical clearance, the game's efficacy can be tested. Sorry. This is the design of our board game. We designed cards. Each color is indicated for a type of cardiovascular disease category, like the light blue is hypertension, the pink is antiarrhythmic, the red is blood forming, and the dark blue is dyslipidemic drugs. We utilize game pieces for movement and a dice as well as a timer. Our game name was Cardio Quest because it's a challenge for us to complete, and these are the basic rules. Is it okay if I go through them? Briefly. Okay. 2 teams or individual players will play against one another. One rolls the dice and moves forward the amount of times indicated by the dice. The opposing team draws a card and then flips it and flips the 2-minute timer. After giving the name of the drug on the card, the first team has to answer the 3 questions. For every incorrect answer, the player moves 1 space back. Then the opposing team repeats rules 1 to 4, and the first team or player to the center or the chest wins Cardio Quest. Thank you, everyone. Are there any questions?
2 questions from the audience.
No one?
Okay.
Okay, greetings to the chairperson, greetings to you ladies and gentlemen. This is Liema. Our topic was to propose a curriculum for advanced wound care for registered pharmacists in South Africa. So this is our poster which was viewed in TV screen tray in the foyer. So basically community pharmacists are the first point of contact for primary healthcare services. Wound care is a basic general management of common wounds, whilst advanced wound care is a specialized field with complex, chronic, and non-healing wounds. So we've tried to review the GPP and SACA guidelines, and we found that there's a superficial information about wound care. The question may arise, should GPP and SACA guideline include wound care as a topic? So there is a limited advanced wound care training for pharmacists in South Africa. Our primary aim was to outline a curriculum for advanced wound care in educational settings and assess the curricula of available courses on advanced wound care management. So these are our objectives that have helped us to achieve our aim. So our study is a traditional literature review. Literature was sourced from online databases. These are some of the key phrases that were used in searches: chronic wounds, wound care management. All literature used was published between 2018 to 2023. So we have tried to review the current BPharm curriculum. What do they cover on wound care? So we have tried to look at the 3 pharmacy schools. So these are some of the topics that are currently covered. So there is a limited number of international and South African short courses identified. South Africa 8 and international 1. So only 3 short courses allowing pharmacists to register were identified, which implies that there's a shortcoming in advanced wound care training for pharmacists in South Africa. Most courses were focused on medical professionals and nurses. So this is the table that summarizes the 3 pharmacy schools. There is a variation in the topics that are covered in wound care in these 3 pharmacy schools. Also, there's a variation in the duration of the topics that are covered in the wound care. So we've tried to propose a curriculum for advanced wound care. The current content includes topics like the types of wounds, the correct selection of wound dressings. So we've tried to propose an additional content on the current curriculum that is covered. Topics like impact on wounding on an individual, taking into account that wounds not only affect the skin but the psychology of a patient, the psychosocial, economical, and environmental impact of the wounds on the patient, understanding the contributing factors to non-adherence to the treatment of wound care. Also, we've evaluated the fact that wound care needs a multidisciplinary approach, both from the pharmacists and the other healthcare professionals. In conclusion, integrating more advanced wound care principle into undergraduate BPharm curriculum will assist in improving the skills and knowledge of pharmacists. Due to its specialized nature, training in advanced wound care should ideally be obtained. Recommendation: it is recommended for South African pharmacy schools to develop training for pharmacists in advanced wound care. Thank you.
Questions for this group? There's Prof.
Thank you. Thank you for your presentation. I can understand the principle of pharmacists becoming involved in advanced wound care because they are dealing with the products in their pharmacy and they sell the products. But what happens when you have a situation where there's a clinic in the pharmacy and there's a nurse professional who is most likely to attend to the wound care responsibilities? In that kind of setting, how do you see or foresee the relationship between the pharmacist and the nurse?
Thank you very much, Prof, for your question. We understand that as pharmacists, we are not really hands-on professionals, but we do work hand in hand with other professionals like nurses. But we are there for advisory purposes. So we would advise the nurses on the correct on dressing and stuff like that. Thank you.
Another one? Another question? People are happy. Thank you. Okay, API, chemistry, I'll call this my miracle group. One day I'll tell you why I call them the miracle group. We're the chemistry people.
Greetings everyone. My name is Jedediah Chabani. I'm accompanied by my colleagues Cassandra and Leona. We were being supervised by Mr. Nzamba. So our project involved formulating a methyl salicylate microemulsion gel for treatment of localized pain and pruritus. Now, what is methyl salicylate? Methyl salicylate, also known as oil of wintergreen, is a compound with a rich history in both traditional medicine and modern pharmaceuticals. It was discovered in 1943. It was discovered in 1943 found in various natural sources, and it is valued for its therapeutic properties, particularly in the realm of pain relief and anti-inflammatory actions. So how does it do it? How does it do its magic? All right. When topically applied, skin esters rapidly hydrolyze the methyl salicylate to the active ingredient salicylic acid, which then irreversibly inhibits cyclooxygenase 1 and 2, so that which are enzymes which convert arachidonic acid which are enzymes responsible for converting arachidonic acid to prostaglandins and thromboxane, which are mediators of inflammation. Now, we then had to choose a dosage form, prioritizing patient compliance, drug efficacy, safety, tolerability, and solubility. We opted for a methyl salicylate gel because it carries advantages such as it is thermodynamically stable, long-term storage stability, and ease of manufacturing. Now we also had to choose packaging. Prioritizing safety and integrity. The packaging that we ended up choosing is an aluminum tube, which served to contain the gel, protect it from spoiling, and provide ease of use. Now, our problem statement. Currently, methods of synthesizing and formulating methyl salicylate have some limitations in terms of scalability, safety, and producibility. So this means that there's now a need to investigate new methods of synthesizing and formulating methyl salicylate. The other challenge is posed by the skin itself, as it acts as a barrier to drugs and foreign materials. We addressed these challenges by investigating different formulation approaches, such as nano-emulsions and microemulsion gels, to deliver the methyl salicylate effectively and safely. Our outcomes were to improve patient care and advance pharmaceutical innovation. So we wanted our project to be well structured and clear. So we established guidelines. We established aims and objectives to serve as guidelines which guide us throughout the project. Now, our aim and objectives, our aim was to synthesize and formulate a stable topical methyl salicylate formulation. Synthesize a stable topical methyl salicylate microemulsion based gel. And then our objectives was to synthesize the active ingredients methyl salicylate and then apply quantitative and qualitative methods to determine purity and identification of methyl salicylate. Then we also wanted to formulate a suitable dosage form for the methyl salicylate and then also test our product. I'm going to hand over to my colleague Cassandra.
The research design was based on mixed research design. It involved both quantitative and qualitative analysis. There was no ethical considerations for this research project since it was solely a laboratory-based research project with no human participants. This is just a table of the materials and equipment we needed and also some safety precautions that had to be adhered to while in the lab. So for the methodology, the first step was to synthesize the methyl salicylate, and then from there, we used UV-vis spectroscopy and trimetric analysis to identify the methyl salicylate as well as to test the purity of it. After this, we then formulated our microemulsion gel, and this basically just involved mixing all the ingredients together and then until the microemulsion was formed. And then from there we did our final product analysis where we tested properties such as the pH viscosity, phase separation, microemulsion stability, and physical examination. And from there we then packaged and labeled the product. This is just the calibration curve for the methyl salicylate and our salicylic acid. From our formulation, this table just shows the different types of formulations we used, and from these results, we had problems where with these formulations, we had lumps that were formed and granules, and also with some we had phase separation. We then decided to switch the gel binding agent from sodium methyl cellulose to xanthine gum. From there, we then obtained the ideal formulation. Our ideal formulation consisted of methyl salicylate at a percentage of 11.26% weight per weight, menthol 3.76, twin 20 and isopropyl at a ratio of 1:1 of 50%, xanthine gum of 2%, and water of 32.98%. Our pH was 5, and there was no phase separation upon centrifuging the different types of formulations. The color was cream colored before the addition of our colorant. It had a medium viscosity. We were unable to quantitatively determine the viscosity of the gel due to the fact that the rheometer in the lab is currently under maintenance. The gel was stable under set conditions in a stability chamber of 25 degrees Celsius and a relative humidity of 80%, and the odor was minty green. This is just a picture of our formulation and the packaging. Discussion, our purity for the methyl salicylate was 94.93%, and for the back titration analysis, it was determined to be 94.58%. The reason for not obtaining 100% percentage purity is due to the fact that some of the reactants could have been lost during the experiment. There could have been fluctuations in temperature. Some of the sample could have been lost during separation and extraction, and also if you used uncalibrated equipment such as weighing balances. The concentration of methyl salicylate was determined to be 0.009840 grams per mole. The concentration of salicylic acid was 4.192 times 10 to the negative 3 grams per mole. From the formulation, as in table 6.1.2.1, when we use sodium
metacellulose as a gel binding agent. The only formulation that was close to the desired properties was when the concentration of metasalicylate was 3%. However, this was very low compared to the therapeutic concentration of 10%. So then we decided to change the gel binding agent to xanthine gum, and from there we could then have the optimum formulation. And then it's just a limitation.
Okay, concerning our study, we had limitations and recommendations, and then in the synthesis process, we experienced some losses along. That could be due to the technique that we use and the UV not including, or maybe, sorry about that, not counteracting every impurities that were associated within the analysis. And also we also encountered that increasing the mass of methyl salicylate in a percentage of 10 grams, it also hindered our product when we're using sodium methyl carboxycellulose. So another thing, time was also a limitation, since we couldn't have enough time to duplicate and validate the results of analysis. So, and also the microemulsion, it was tested under 25 degrees Celsius and 80% humidity only. So, and also viscosity, we couldn't determine the viscosity because the rheometer, it was under maintenance. So in conclusion, in short, this study, it was performed and purity was assessed using both percentage and trimetric analysis. Overall, it was 94.76%. So furthermore, what matters here is that it needed meticulous experimentation, and also adjustments were made based on were made based to counteract, to make sure that the product it is stable and safe. So here are the references, and we'd like to say thank you to Mr. Nzamba, Ms. Arista van Jasveld, and Ms. Jean van Jasveld, and NMU as an umbrella.
Any questions? Prof?
Thank you for your presentation. I think you've got a potentially good product over there. And I wanted to just ask you, in your studies or in your research and literature review, I understand this to be a topical product. I mean, it's for localized use as an anti-inflammatory to get into inflamed joints and muscles, I suppose. Were there any concerns about, you talk about improved bioavailability. Were there any concerns about systemic toxicity from the ingredients of methyl salicylate and menthol?
I did look into that, and they found out, like the literature that I read mentioned that when it's applied topically, the concentration is not that high that it can cause systemic side effects. So that concentration is fine for topical use, it won't cause any side effects.
Thank you very much. Very interesting presentation. You very nicely explained the mechanism of action of methyl salicylate as an anti-inflammatory, but you referred that it also has antipyretic properties. Did you find out how it works against itching, what its mechanism is? I'd be interested. Thank you.
Yes, we did look at that as well. There was no definite scientific reason for that, but upon discussion with our supervisor, he did mention that because of the cooling effect that the gel provides, that will help with itching if you apply it onto the skin.
One last question. Is your voice loud enough?
Yeah.
Okay.
that instead of the method of cellulose, you rather opted for something gum. I don't know why you did.
Okay. At first we use carbox. Okay, in short, what matters is that we also use this product for musculoskeletal problems, as mentioned, joints. So, you know, we also consider athletes, and athletes, they get sweat. So, in short, methyl carboxycellulose is not suitable in the presence of salt. So we opted for xanthine gum.
So, chronic wound poses a global health crisis affected by both local and systematic factors, such as foreign bodies and diabetes, respectively. The main problem with the available treatment is that it is ineffective to treating these wounds, which then leads to them developing infections and pain, resulting in severe consequences such as leg amputation in patients that have diabetic foot ulcer. So, liposomes are a spherical vesicle made of phospholipid. They are a promising drug delivery system for chronic wound treatment due to their biocompatibility and biodegradability. Aloe vera gel accelerates chronic wound healing by promoting angiogenesis, collagen deposition, and reepithelialization. Biogenic copper nanoparticles exhibit potent antimicrobial and antioxidant activity, promoting chronic wound healing. Ibuprofen is a non-steroidal anti-inflammatory drug and has analgesic and anti-inflammatory properties, which then may lead to it promoting wound healing by reducing inflammation and pain. Incorporating both copper nanoparticles and ibuprofen was to aim to enhance wound healing and pain relief, capitalizing on their antimicrobial, anti-inflammatory, and anti-analgesic properties. These are the methods and the material we used. We first pick aloe vera leaves. We first pick the aloe vera leaves, these ones, from Mission Vale campus. We pick both dry and fresh leaves. These leaves were then broken down into small pieces and boiled at 80 degrees for about 15 minutes. This was then mixed with copper 2 nitrate trihydrate solution. Formation of copper nanoparticles was confirmed by the appearance of this brownish to black color precipitates in the solution mixture. Phytochemical metal reduction and capping was done. Encapsulation of liposome and ibuprofen was fabricated through a microfluidic system. Then, the optimal liposome formulation was then integrated into an emulsion cream formulation.
Copper nanoparticles and ibuprofen were successfully loaded into liposomes with a small diameter and high entrapment efficiency. Then the liposome were incorporated into a cream formulation. The first picture is showing copper nanoparticles. The second picture is our zeta potential that was minus 20 millivolts, and it was within the targeted range. And the third picture is the optimal liposomes exhibited high ibuprofen entrapment efficiency at a percentage of 89% and sustained a drug release profile over 24 hours. In conclusion, liposomes loaded with copper nanoparticles and ibuprofen were successfully synthesized for chronic wound healing and pain management. The physiochemical properties of liposomes were characterized, confirming its suitability for targeted delivery. The in vitro release profile demonstrated controlled release of copper nanoparticles and ibuprofen. Further research is needed to assess the antimicrobial efficacy, stability, and safety. And this is our poster.
Two questions. There's Mr. Zalu over there.
Hi guys, and thank you very much for your presentation. So I just have two questions for you. The first one, I would like to know what method did you use for your encapsulation, did you use an active loading or passive loading to encapsulate your ibuprofen and also your copper into your liposomes? And secondly, you have stated on your conclusion or your findings that your product or your formulation had high encapsulation efficiency. So I would like to know, how did you calculate that? Thank you.
Okay, thank you for the questions. The first one with the loading. So we use the passive method. And then with determining the efficacy as to why it was higher than the others, you mean why was it high, or how did we know it was high? Yes. Yeah. Yes. So we use the techniques like your UVVs to analyze the thinking. Do you mean that question? I don't think I get where you're leading me. But I think it's one thing we can look further to it, but then it was in that percentage when we determined the efficacy of the drug. Oh, that's the question. How do you calculate the question? Put it right. No.
So sorry, they didn't calculate the system. The system calculates if you all. You've got fancy machines, they do everything for you. So, one last question. One last question. Yes, Sarah, yes. Game-based learning, last group, get ready.
Thanks, it was fascinating. It just pure curiosity, please educate me. Why did you use aloe vera? What was the role of aloe vera?
Aloe vera, we did a literature review, like aloe vera showed like good results, and we check the properties of aloe vera. We found that they are active ingredient that can actually help in our research, and it is a good extract to use, because at the end we get what we want.
So to add on to what he was saying, it has antimicrobial effects. So it helps with wound healing to stop, so it kills bacteria that can cause infections on the chronic wounds. So it has antivirals and antibacterial properties. Yeah, we did.
No, it doesn't.
No.
No, it doesn't.
Thank you. Okay, are you ready? I'm starting.
Good afternoon, everyone. My name is Asipila Nongoba, and this is my group. Our title for this group study was the development of a game-based tool for pharmacy students to revise over-the-counter pain medication directions for use in adults. The background: game-based learning and gamification refers to the borrowing of rules and principles to engage users and applying them to real life. It can be in the form of flashcards or computer software to engage educational material in a dynamic and playful way. It is evident, we all know that second-year BPharm students are required to do work-based learning in pharmacies, and they are exposed to active pharmaceutical ingredients, and they get overwhelmed with the numerous generics that are used in the pharmacy setting, especially for adult pain medication, and this can be difficult and pose a threat to giving the exact directions for use and counseling to patients. The aims and objectives. The aim was to develop a game that enhances student learning to understand medication directions for use. And the objectives include adapting a learning tool which could be used to recall medication directions for use in a pharmacy, and also to create a gaming tool using the MDA framework to assist the development of the game while bridging the gap between game analysis and game research, mechanisms, dynamics, and aesthetic framework. So mechanics include the rules and codes. Dynamics are is basically the runtime behavior while playing the game to aid in maintenance and creation of aesthetic experience. Aesthetics is the response to the game, including the fun aspect of the game. So the game was created by the group 3 of the group study project and using an existing game called 30 Seconds. I think we all know the game. PRX, the name of the game, is a non-digital game. PRX originates from the word prescription, which is mostly referred to as RX. The game includes different types of over-the-counter pain medication names with pictures for clarity to state directions for use, maximum dose, cautions, side effects, and counseling. The game mechanics were made evident by creating game rules to help guide and control players' behavior, including the timer, which is 60 seconds. In application of game dynamics, the game context is based on dispensing over-the-counter pain medication appropriately. The player is given a variety of pain medication cards to play with. The game rules are included in the poster. In conclusion, the objectives of the study have been successfully met through designing the non-digital game PRX with an intention to refresh knowledge when reviewing over-the-counter pain medication directions for use. It is unfortunate that no feedback was generated through experience of applying the rules and playing the game, as no ethical applications were made as a result. No players played the game and no data or results were obtained. In future, if ethical applications could be made, the generation of the data and the feedback would be possible to enable the researcher to improve their researching skills. Non-digital gaming evokes interpersonal interaction and is important in raising awareness. PRX should make players aware of the dangers of incorrect pain medication directions for use. These were the references that were consulted, and thank you for listening.
Two questions? There's a question over there. Is your voice loud enough? Okay, let's give it a try.
How would you convince me that the game actually works on students, since we didn't do any real-life experience?
Can you hear me? Oh, so this game was basically done as a module for pharmacists. We were not doing this to test. Okay, so if we are to do it, like in the terms, let's say we're going for the sponsorship, then we would need to do further research and actually put the game out so that we can test using our participants. But in this case, we're just doing it as a model for the module.
One more question. Advanced wound care, last group for the day. Please come forward. Thank you so much, guys.
Good afternoon. afternoon, ladies and gentlemen, and a special warm welcome to our chairperson. I just want to say, if you're still here, thank you for the support. We are Group 1 of the Advanced Wound Care Elective, and our topic was the identification and classification of wound care products in community pharmacies of South Africa. So this is our lovely poster that we designed. So historically, the wound care products that were used to clean wounds was actually things like milk, water, and even vinegar. And then topically, you would apply honey or any occlusive dressings that would provide a good wound healing environment. Due to newer advancements and products being developed, we found the need to classify these products and make it part of our research. So our primary aim was the identification and classification of these products in the community pharmacies, and also to identify which companies supply these products.
The methodology chosen for the study, The methodology chosen for the study was a traditional literature review. Searches for articles and websites were conducted using the databases listed above, like Science Direct, Google Scholar, Wiley Online Library, 7 African journals. Keywords such as wound dressing South Africa, wound dressing, wound products classification global, and wound products classification FDA was utilized. Information was gathered, categorized, and summarized using Microsoft Excel. So, the database search identified 25 papers in total, and following thorough evaluation, and duplicate and 11 articles were removed, and 12 articles were deemed relevant. Inclusion of research articles were limited to those published within the past 5 to 7 years. Articles not relevant to the categorization of wound care products in South Africa and internationally, wound care products not intended for human use, and articles not peer-reviewed and not published in English were excluded to maintain the research precision and reliability. Data was analyzed in terms of general overview of classification internationally and in South Africa.
Okay, I'm going to discuss the results in the discussion with you. So we started with an international classification. As with all research, you start big and you work your way to your smaller points. So we identified the FDA, which is the US version of like our SAHPRA, that's called the Food and Drug Administration, and they classify according to risk levels. So with class 1 being your lowest risk and class 2 and 3 being your intermediate to high risk. Australia focuses on sterility, and the European Medicine Agency is a regulatory body for their wound care and their burn dressings, and they are in a class 2, so that would be an intermediate risk. Then we can look at South Africa. So we are classified by SAHPRA, which, as I'm sure you all know, is the South African Health Products Regulatory Authority, and they use a system of class A through D, with class A being low-risk devices and D being a severe risk. It is also categorized further into 2 groups based on constituents for and intended uses. This can be seen as the table provided on the screen. Furthermore, South Africa also has the Wound Healing Association of Southern Africa that also has a classification system. The dressings are classified according to their properties, such as moisture control and antimicrobial properties. We also identified 11 of the prominent companies in South Africa that do participate on the market, and they can also be viewed by our figure that we've posted there. I'm going to hand over for the conclusion.
In conclusion, we were able to see what classification systems are used in South Africa. We were able to compare SAHPRA with other countries like United States of America that uses the FDA classification system. We were able to find that there is no universal classification system for wound care products. However, there is a parallel between SAHPRA and FDA. The limitation of this study includes the lack of first-hand data regarding the utilization of different wound care products available in South Africa. Another limitation is the researchers' sole method of gathering the information was internet literature searches, which might have not allowed for comprehensive understanding of the current market situation in South Africa. To improve our study, ethical clearance must be obtained for further empirical study on the table. Thank you.
questions? There's Ms. Tsalu.
Thank you very much for your presentation. I have two questions for you. So my first question, I'm sorry if this might come as a bit offish, but I kind of found your study a bit, or your title a bit misleading, because your title, you say that you were going to identify and classify products in community pharmacists, yeah, right? Whereas you have conducted a systemic review, because for me, judging by your title, I thought you were going to go and conduct the study and identify the product in the pharmacies, and only to find out that you only conducted a literature review. My second question I would like to ask, that was a comment. My question, so when you're doing the classification, I would like to know which wound products were involved in those classification system. Were they only including dressings, or there were other wound products that were part of the classification system that we have identified? Thank you.
Thank you for your question. So basically, we looked at SAPRA's classification, and they classify wound, I would say, wound dressings. And there's also other things like, we actually had another table. They're classified based on the constituents and the intended uses. So if they are used for like bandages, then it's going to be another one. And dressings like your antimicrobial dressings, antibiotic dressings. So it's a big range that was conducted, I would say, included.
Okay, just to add to what my colleague said, you said that our title was a tad misleading. So yes, I agree with what you're saying. And we actually started with that, and when we started doing our research, we actually realized that to fully satisfy that specific title, you would need that ethical clearance to actually go into the pharmacies, see what the recommendations are by the pharmacists, see what products are bought in, how the sales go, and then also with regards to what products and how they are classified, we have much more depth in our report about that. Thank you.
Okay, thank you very much, guys. This wraps up all the presentations for today. A big yes. Okay, I'm gonna call Dr. Kubashe. I really feel so bad because I threw it right under the bus today. Dr. Luvuno was supposed to be doing this, but because she fell ill, I had to hijack someone. And poor Dr. Kubashe, with her limping leg, she agreed to come and talk about the Clinicare Award plaque.
A very good afternoon to everyone who has stayed up until this time. And I would like to extend a very warm thank you to the director in her absence for research and Prof. Naidu. I want to thank Mrs. Alice for gracing us every time. Our judges, all protocols observed. I want to apologize for coming onto the podium very semi-casual or casual. I blame Dr. D, and maybe I will conclude that with the teacher that is likely to prank other teachers. Okay, just to get to the business of the day, I would like to really thank you, Maxine, being our alumni and coming back here for that, and give an inspiratory message to our students. It really means a lot. That means the Nelson Mandela Pharmacy is present. We are known out there because of the alumni that is out there. I thank you very much for that. I love your opening statement when you said your research journey started with your love for the people. And that is what pharmacy is all about. Taking us back to first year, a pharmacist, the caregiver, the pharmacist, the communicator, the pharmacist, the researcher. So thank you for that. It was beautiful listening to that because it all went a full circle today. And we have research today because of the heart of the pharmacist. Now, when we come to the plug, during my days as Prof. Naidu, as one of my lecturers, and my HOD at the time, this day was a symposium evening sponsored by Aspen, as always, up until today. And then it was more about laboratory. And it was just laboratory that you would see. I think the first presentation or maybe project that came out as a clinical project was one by Bianca, the late Bianca, and may her beautiful soul rest in peace. She was one of our students as well. So with ambassadors like Maxine, they went out there and they identify the importance or maybe the gravity of the clinical pharmacy that you live with when you leave our premises. How much we drill you with the 0 criteria that you so not like, and what it can do out there. Nothing that we do here cannot be translated into practice. Everything we do in our labs translate to doing something for the patient on the outside. So this year, she has been very instrumental with Clinic Care to sponsor us with another plug, because the plug that we all had was the pharmaceutical sciences one, and now this year, 2023, we are introducing a new section, which is the clinical pharmacy plug. And thanks to clinical pharmacy and you, Maxine, for instrumenting this one for us. We are very grateful. And now we know that whatever that you are doing matters outside. Thank you very much.
It's heavy, guys.
So if it is a clinical pharmacy project that wins today, you will be the first person to go onto that shield.
Thank you, Dr. Kubashe. Thank you for the short notice, the way you spoke. You'd swear I asked you last month to prepare a speech. Thank you for that. And thank you so much, Maxine. I don't know who made that wood, but that thing is very heavy. I doubt if I'm gonna need to go to gym this week. But I cannot wait to see the names being populated there. I know we've got few third years that are listening online. The challenge is on. You better prepare next year to have your name there. Okay, the moment we've all been waiting for. Are you guys ready with the totals? Are you ready? Okay, so we're gonna call Mr. Nkomo to come to the front, and we're gonna start with the ZRE groups and then the ZCE groups. So he is the drum roll person. Let's give him a hand.
ZRE first. Ladies and gentlemen, good evening. I'm here to deliver the results as you have been waiting. I've got your top 3 here. And if I call your name, please do come to the front. If I don't, you still did well. Okay, I would like to put a disclaimer that I was not involved at all. So if you find yourself not coming to the front, I am innocent. I was not involved in putting together these. I'm just a messenger. Having said that, from number 3, we have student Musekene and Sibanda. Well done, ladies. Run, 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 run. Well done.
Okay, Mr. Ngoma wants to know what are they getting. We still need to. This is what they'll get. They'll get a certificate and a gift. But the administration in organizing all of that, it's a long one. They still have to write exams. So by the time they finish the exam, they will go home with their certificate and a gift. So we're not giving them anything today, we're just showcasing them.
Okay, going to number 2. Would you like to take a guess? Also, another disclaimer, I am not involved. May I please call to the front Miss Mutsunguma and I told you beginning of the year that you should come and join my group. Number 1. The winners of the 2023 ZRE 400 Podium Research Symposium. Okay, Casey and Hope. Well done, ladies and gentlemen.
Go meet her halfway.
Okay,
are you ready?
ZCE number 3. Drum roll, please. Mr. Kenoga and your friends, please come to the front. Number 2. Okay. Buthelezi L et al.
Well done.
All right. Thank you so much for coming. So apparently we do not have number 1, unfortunately. No, I'm kidding. The Miracle Babies, Zimtal et al. Number 1.
Good afternoon, everybody who's here. I know that Mr. Gomo has just said good evening. I looked at my watch. I think we're still in the afternoon. I'm not going to waste anybody's time. We are way over time at the moment, and I'm not going to try to repeat what has already been said, but I would like to thank our guest speaker, Mrs. Tromp. She was one of my students in one of the first ZCE project groups in 2015. We did a cold chain study together with Adcock Ingram. And today, the message that she really put out there is that all of you guys need to come back next year and the subsequent years and join us in our research. Here's Dr. Tambuza. You know who to speak to if you want to further on. Thank you very much, Maxine. And I would also like to acknowledge and thank our parents, any parent who is here, for encouraging the students and making sure that they are motivated, they are able to deliver what they have delivered today. We are very proud of what you've actually achieved today. In fact, let me also add by saying that we have announced 3 winners for ZRE. And then we've got also top posts for ZCE and winners as well. Today, I just want to say that everybody who's here is a winner, and we all deserve a round of applause. You are all winners in your own rights as you sit in those chairs. And also, without the involvement of the marketing team, we would not be able to have all the lights here. the music and everything that we see right in front of us. Thank you to Mrs. Berry and Nelisa and all their involvement, in all the activities that we always have and events in pharmacy. They are always, ready, available, and willing to assist. Let me also take the opportunity to thank our encyclopedia, Prof. Emeritus Naidu. Thank you for being here and always, engaging with our students. We learn a lot from you each and every day. PSSA is represented by Ms. Alice Latakhan. Thank you very much for being here, ma'am. We're always happy to have you amongst our midst. And our colleagues that are here, all the lecturers that are responsible for nurturing these students, because at the beginning of the year, January, February, the students do not know what a research proposal is all about. So they have to be taken through the entire process. Thank you to Dr. Dambuza for being the coordinator of the ZRE and the ZCE module. She does a tremendous amount of work. We all follow after her. I think she also deserves a clap. We have got external supervisors, Dr. Mpo. We've got external supervisors also from the biochemistry department by the name of Donay Kitzinger. Thank you very much. I'm not sure if she's still here. We appreciate and thank you. We also initially were embraced by the presence of Prof. Melariri, from the research directorate. She didn't come alone. She came here with an entourage of her people, including our judge today, Tembeka Sitinane. We also had Zukisiwe. We had, what is the other person's name? Bethany Morton was here also. And we also had Lutando. I'm not sure if they are still here. Thank you very much for the support and the work that you do for our department. And the judges themselves, Dr. Rian van der Venter, thank you very much. We appreciate the work that you've done, putting aside everything that you had to do and coming and spending the afternoon with us here and our students. And also you, Mr. Jinane, as a judge, we thank you very much. And we are giving you a gift now to say that we appreciate your presence and your judging, of today's event. Who else am I missing? The leadership of Dr. Luvunokile in her absence. We thank the madam very much, and we appreciate always being guided by her. I hope that she recovers well in her minor ailment. Our sponsors, Aspen Pharmacare and Clinicare, once again, thank you very much. Things would not have been, this beautiful without you. We thank our horticulture department for all these beautiful flowers that we all see in front of us here. But also, we would be in the dark. Our sound and voices would not be heard if it was not for Zukile. Thank you very much, Zukile. Initially, Philip wasn't here, but I see you here now. Thank you very much, Zukile. I was told that you were somewhere else in another event. It's always good to see you. Thank you very much. If I am missing any person, that is not done deliberately. Just in closing, just to thank everybody who's here, the support, and thank you for your time. It's always great for us to see the students coming in at the beginning of the year and being taken through the research process I've actually indicated, and now coming up with final research projects and posters and ready to present them, and most of them can be developed into an M study and all the way to PhD. So we really encourage you to come back. We will supervise you in your numbers. Thank you very much. Thank you.
Colleagues, students, this is it. I don't think there's anything else that we haven't done. Students, you now must go and prepare for your exams. We are going to be, I'm still talking as a belief. I haven't dismissed the class. You're going to be writing your exams, and within 6 months, in fact, even less than 6 months, you're going to be wearing a black gown, walking across the stage. That is my prayer for you, that all of you get to be there. I will be sitting there on the stage, and I'm going to be looking for each and every one of you on that stage. I wish you all the best, and go on, take a nap, and then study, and make sure that you give your best. This is the last stride. Give it your fastest, like Kipchoge. I know some of you don't know who that is, but give it your last stride and break a record this next couple of weeks. We can't wait to see you graduating. I wish you all the best. Colleagues, drive safely. When you go home, it's very late. It's almost 5 o'clock. And yes, we went over time, but we were having fun, so who cares? You know? and yeah, I can't wait to have this, you know, gosh, can you see how beautiful it is? And I'm sure everyone now wants to change their projects, but I can't wait to have that displayed in our department. Thank you so much, Maxine, and to your company. And Prof, it's always a pleasure to see you. Thank you, colleagues. Have a lovely evening. I now, by the powers vested in me, I now declare the meeting dissolved.